Hello. Poonam.
Yes, ma'am.
Hello.
Good
Yes.
afternoon, good morning, and good evening to all present here. I, Poonam Daya, Assistant Professor in Computer Science and Engineering at Jagannath University, Bahadurgarh, India, and co-convener of this webinar, welcomes you all on the second day of International Webinar on Deep Learning for Control and Management of Plant Disease. Today's session is in continuation of earlier session held on 8th March 2021. First of all, I would like to invite Professor Sumer Singh, Vice Chancellor of Jagannath University, for formal welcome of all the speakers. Professor Sumer Singh had been selected as engineer by Union Public Selection Commission, India, and he had been Vice Chancellor of Sangam University and Career Point Kota University. Please, sir.
Thank you, Poonam. It's a nice introduction and the start for the international webinar, and that is to the second day and the second session. So I think the topic which has been selected, it's quite apt because since last so many years, maybe the computerization and then we have gone beyond the computerization and the cloud technology, many advancements in chip technology has helped us to go ahead in all these areas, almost for applications of deep learning, artificial intelligence, and many other newer technologies in every field almost. So it's my proud privilege to welcome speakers, delegates, and all the academia fraternity members present here who belong to not only the national universities, the institutions, but the institutions of great repute at international level. But let me tell you, I think whatever change is happening, that is going with a faster pace. Even in today morning news, I could see, and let me share with the pride, with all the even the foreign speakers, that India and the Indian education, I think they are going in a very faster pace as far as reforms are concerned, as far as induction of technology is concerned, and even the interdisciplinary approach. The news of today, which has been released by AICT, that even in the year 2021, the students who will take admission in BTech even need not to have the physics and mathematics as subjects in that 12th grade. So I think people may take it as surprise, people may criticize, but to my mind, this is where the future is going to lead us, all the academicians and the learners of 21st century. Because today, every discipline has intruded to another discipline in such a manner that yes, it's very difficult to distinguish. And the same way is happening even in the agriculture science. Somebody who has done 12th agriculture, even they can go for BTech programs. So this is the recent news which I can share and we can think of. And the background which Professor Bahal took as chair of the webinar, that he is always forward thinking and always try to plan the deliberations at international level which are meaningful and taking us to the future, which is looking forward for the newer thinking, newer concepts, and use of technology, and that is to advanced technologies like deep learning. So, with all these things, I'm looking forward for great deliberations. Let me express heartfelt thanks to the overseas speakers of today who have spared their valuable time and making the deliberation highly learning and meaningful for all the participants who are participating in this webinar across various countries and their institutions. Thank you. Poonam, over to you.
Thank you, sir. Let us start today's session with presentation by Dr. Surya Kant. Dr. Suryakant has over 20 years of international research experience. He is working as a senior research scientist and leading a research team at Agriculture Victoria based at the Grains Innovation Park, Horsham since 2011. He is also principal fellow with the University of Melbourne. Previously, he had research scientist positions for 12 years at the Department of Molecular and Cellular Biology, University of Gulf, Canada, the Department of Dryland Biotechnology, Ben Gurion University of Negev, Israel, and the Faculty of Agricultural, Food and Environmental Quality Sciences, Israel. I would like to invite Dr. Suryakant to present his views on modern phenotyping, analytics, and artificial intelligence for crop breeding traits. Please, sir.
Thanks for the intro. So, one second. Yep. Can you see my screen? Not yet. Okay, so it is no. So for some reason, I have to take a, because this is my work computer and this is not allowing this website to share my screen. So is it possible that can take start over and I have to switch my computer actually? Looks like it's not allowing me to share my screen.
Try again, sir, please.
Dr. Suryakant,
Yep.
you can send your slides to this side and maybe we can try this project.
Sorry, I didn't hear that.
I'm saying that you send your set of slides, we will try to place it. You may send your PPT PowerPoint presentations to us by
No,
WhatsApp.
it's 140 megabyte. It's too large file to share.
Yeah, maybe that is the reason that it is not coming.
Yeah. No, it's just blocking my screen to share. So I would have to, 1 second. No, I think I have to change the computer. It's because of my work computer. It's not letting me to share the screen.
So can we have Dr. Phenomics first? He will take
Would you do me that favor to to broadcast your presentation first?
Yes.
Yeah,
Hello,
Tino is happy.
Can you help us by being the first speaker?
Can you hear me now?
Yes, yes.
Yes, sir.
Okay. So sorry, I have no webcam attached to it, but it's not about seeing my face. It's more about the content that I have to share. And please don't do a formal introduction. I mean, I could just introduce myself very, very briefly. So I've been working for 10 years in the scientific environment, doing my PhD and postdoc on plant phenotyping in general. So I've been working on all kinds of aspects of application of sensors for monitoring biological objects. Then I've been working also for Lemnatech in the past 5 years, and now I am working as a freelancer and also associated to the Lab under which flag I will going to present now a little bit of my viewpoint. Let me see whether I can share my screen. I think the browser has blocked your screen. Click share icons. Oh my god, this is always the same thing when things. Okay, share screen. The browser has. Click the screen icon and then you address the bar.
So it looks like mine is. Can you see mine now? No.
Nope.
I think you should be able to see now or no?
Yes, sir.
Yeah, I got it. I had to unblock the firewall. So can you see now?
Yes, sir.
Yes, sir. It's
All right.
visible.
Sorry about that. Yep. So thanks a lot to everyone who has been organizing this nice program, and special thanks to Dr. Bell. It's been like 25 to 30 years ago since we met, and he has been our teacher, my teacher, and it's been sort of a good inspiration for my career to move forward. So thank you very much, Dr. Bell, for organizing. And so, yeah, I'll show in that theme some of those modern phenotyping technologies and what novel analytics and artificial intelligence approach we are applying for important crop breeding traits to digitize that whole approach. So at our institute, basically, we are sort of aiming for accelerated precision breeding for grain crops. Those grain crops include wheat, canola, safflower, lentil, and field pea. And we are combining all the different spokes in the hub, computational breeding, genomic selection, genome editing, genomics, and in specific, I will show more about this phenomics. And overall, those approach are sort of linked together for the germplasm enhancement for the better elite varieties in these grain crops. So that is sort of overall narrative and aim for our institute to focus on those crops. And my group as such is involved in this phenomics. So we have recently established a phenomics hub we named Smart Sense iHub. That is a key research as well as entrepreneurial. I will show in the last what I mean by entrepreneurial, but commercial or applicable things we are doing. But overall, we are trying to provide innovative solutions to agriculture industry and giving novel analytics, digital phenotyping, and predictive modeling in this space. As most of you are aware that phenotype is whatever the traits we can observe from outside, whether it is growth, biomass, morphology, physiology, agronomy, and ultimately the yield. Traditionally, this phenotyping has been done through manual approach, taking the manual notes or doing the measurements, or a lot of time destructive harvest. And that is all become laborious, slow, and less reliable because it change from the person to person who is taking the observation. And for that, we are replacing this with the digital phenotyping in control environment glasshouse condition, where this automated plant growing system and imaging system is there, and in the field using drones and different sensors. So to make it more automated, non-destructive, and more reliable. So here's a list of wish list kind of things which a plant scientist or breeder want to capture in their trial, and what are those different traits and how those measurements are being taken. So for example, growth, it can be measured in terms of biomass, leaf area, yield, morphology, height, architecture, phenology, photosynthesis with gas exchange, pigments, by spectral signature, plant water content, different biochemical components, protein, carbohydrate, and stress level through canopy temperature. Obviously, we are focusing more on the disease. So how the disease resistance, severity, or it can be estimated. Most of the time it is done scoring, and root growth and grain quality parameters. So those are the list of things which are sort of you wish to have comprehensive data set to make some conclusion for moving forward specifically in terms of breeding, if you want to select the genotypes. But again, most of those observations have been done manually. So from that wish list, we narrow down to the list which we can do through the imaging. So these are the traits here from the previous list, and these are the imaging technology, RGB, multispectral, thermal, hyperspectral, and LiDAR we are using. And what are those different index or measurements you need to capture through those imaging to draw those trait data output. So status here, we are showing this, we have been able to do all those in multiple crop species. And some of the others we have established protocol and analytics in 1 or 2 crop species, and what we are trying to apply because sometimes the morphology of a monocot and dicot change significantly, and you have to change your approach and analytics based on those crop types. For the glasshouse control environment condition, we have this 2 plant phenomics facilities. Both have LaminaTech system. One is at Horsham and another one is at Bandura. So just to give you a little bit brief, most of you are aware, there is color imaging system where we do top view and side view. Hyperspectral imaging, we have range from 550 to 1700 nanometer. That we have been able to do some biochemical parameters, specifically chlorophyll, which I show later, and some other vegetation indices as well. And it's all automated watering and weighing there. And this is sort of like just a brief, like what are the different traits in that glasshouse phenotyping facility we have established. Nitrogen use efficiency, salt tolerance, water use efficiency, and disease resistance. And in those protocol, we have established and validated phenotyping for thousands of accessions, mainly for the wheat, but we have done those correlation studies for wheat, lentil, chickpea, field pea, canola, and safflower. These are a number of different publications underlying those phenotyping approach we have been doing not only in this facility, but previously as well. So this is just an example of a digital biomass and growth rate. Shown here is raw images on the upper panel and process image grown at different nitrogen level. And cool thing is that automated facilities that you can image at really frequent interval, and that give the growth rate, and that give better confidence to draw your conclusion that of which genotype is performing at what growth stage in a better way. So this is a sort of a layout and installation of a hyperspectral sensor system. Won't go much detail, but this is sort of a really like novel thing which we have developed is using the MATLAB. We have been able to nicely estimate different parameters. I will show one example for chlorophyll, but here it is the approach which we have done for the segmentation classification and how different parameters in the field of view, for example, the background cage, plants are grown in cage in this example, pot and soil, all those background can be nicely differentiated, and in the green line is the plant as a foreground that you can capture to estimate your different vegetation indices. For chlorophyll, we used his brute force computation approach in MATLAB, and we were able to get this novel vegetation index for estimation of chlorophyll content with a really good high correlation. That was repetitive few times we have done. And for the field condition, we have different aerial platforms ranging from small to very big, and a short flight duration to the long flight duration, or different payload capacity, because those sensors, they come in different weight and carrying capacity. So, and then sensors, we have digital RGB cameras, multispectral LiDAR, thermal, and hyperspectral camera for different traits which I was showing to estimate. And again, for taking those images or data is easy thing, but important thing is how you can make.
Hello, sir, Dr. Suryakant, sir.
Hello, yeah.
Sir, sorry to interrupt, but your slide are not moving. We are just seeing first slide. Now it's moving.
Oh, really?
Kindly check.
Sorry, thanks for telling me. But
Okay,
maybe
right.
now it was in the slideshow mode, so I have to sort of maybe skip that one. Slideshow mode. All right, so I will just go a little bit here. So these are the different aerial platforms, ranging from small to big one, with the different flight time and different payload capacity, and different cameras, RGB multispectral LiDAR thermal cameras, and hyperspectral, which we are using for different traits, as I was mentioning previously. Sorry you missed that one to see. But those aerial phenotyping data acquisition is really easy, but how to analyze, how to process the data in a sensible way is the key thing. So you start with those image acquisition and different UAVs and sensors, and then we process those images mostly in the Pix4D software and analyze those images to have the estimation at plant level or the plot level in different GIS software or the object-based identification software. And then we estimate those phenotype for the examples which I was showing. Probably maybe it might be worth having a look again for those for sake of, because these are the different traits I was mentioning. Seedling emergence, early vigor, biomass in this left is the traits, and then this imaging platform, RGB multispectral thermal LiDAR, and how those sensors and cameras, they translate into different measurement is shown here, and then the status for those observations. Again, so that's the important thing. And then we are able to tell the even farmers that how those data set can be useful for the farm package allocation, for example, input application or control of different disease. And this is just an example of how we do the flight mission, automated flight mission, and a ground control point, and where we can have accuracy of less than 1 centimeter, and different calibration targets you have to obviously use. And once we acquire those images, there are thousands of images for a trial of 1 hectare site. Generally, we have 1,500, depending upon what overlap you are doing and what speed you are flying. And overall, it looks like this. This was like 1,800 field plots, 5 meter by 1 meter size. This is composite image, orthomosaic image for RGB. This is for NDVI. This is for the thermal camera. And as I was saying, that this is how we apply that analytics. These are 5 meter by 1 meter plots for wheat in this case. And we draw the polygon in the GIS map, and then from those polygon, we can exactly overlay the field plots of that site And from there, we can really estimate, okay, which genotype in which plot is behaving in terms of growth and biomass or growth rate. This example of seedling emergence count, we use e-cognition software, and this is sort of in the field at early emergence stage, and we could classify the plant, and again, but when we classify the plant, it can pick the weeds as well. So we have provided input as a more than 1,000 images as a template, and from those template and overlay, we were able to count exact number of plants in the field conditions. And similar approach we could apply for the flower count. And this is sort of like an interesting phase we are going. We have phenotype more than 6,000 wheat accessions. They are worldwide collection, including varieties, land races, and breeding lines. And then those we have phenotype in glasshouse as well as under field condition for yield, quality, and 3 different rust resistance. And as you can see, the diversity of those reference population range really humongous. Multiple spikes, spindly spike, different maturity or phenology, and different plant height. And that we have recently published a data set in Nature Genetics about the diversity of this population and how it delineate different traits in this population. So here it's sort of like diversity. You can see on the right side is high nitrogen and low nitrogen that field trial growing. And again, in the aerial footage, you can clearly see the diversity of the 600 wheat reference population grown in 3 replicates. It's really interesting and amazing. And from that diversity panel, we were able to screen for different rust resistance genotypes, and what are those different QTL linked to those one. So that has also been recently published in TAG journal. And yeah, this is another example of in disease space, Russian wheat aphid. A couple of years ago in Australia, it came from overseas, and since then it has keep growing. And Grain Research Development Corporation, which is a main funding agency in Australia, they have put a rigorous emphasis to control this one, but as well as to develop Russian wheat aphid resistance genotypes. So this is how sort of we grow in a single row to do the disease severity or estimation. But again, this is like 50 centimeter row, but from the aerial images at 25 meter height, we can really go to the good resolution to differentiate the control or resistant and susceptible genotype. And on this right, here we are showing the hyperspectral imagery data that how those control and Russian wheat aphid signature are differentiated. That has been helpful to do the high throughput phenotyping using this hyperspectral camera. And this is another example of field P reference population. We have about 1,500 accessions grown in the field. And the idea, there was a bacterial blight we were trying to study, and that we were trying with the decay in cropy greenness, because bacterial blight at sort of a post flowering stage, it really become challenge, and you can see the really wilting and disease symptom. And instead of taking scores, we were able to estimate how the decay in crop greenness was correlated with this bacterial blight score. So that was quite efficient, nice approach. And here there are 3 other disease example: bacterial blight in field pea, botrytis gray mold in lentil, and escogita blight in field pea. Again, this is thermal map. This is much more clear where we were targeting, and nice gradient of that heat map. It clearly show that this side it was sort of a control, and this side it was like treated. But clearly you can see from your naked eye that In the orthomosaic image, there was clear demarcation of that, but when you analyze the data at a plot level, it clearly shows sort of a nice differentiation for the resistance and sensitive genotypes. And now moving on to the next phase, like we have been applying more recently this latest technique of this deep learning, machine learning, automated machine learning, and this is sort of like recent wave in artificial intelligence, and is quite high performant machine learning pipeline that can be applied with minimal user effort. And there are major companies, Microsoft, Google, and Amazon, they are offering this, but at a really high price tag. So that's the challenge, that most of this commercial solution by these companies, they comes with the significant cost. And one of the issue for us is that the sensor-derived data, multispectral, hyperspectral, or LiDAR, which we want to analyze in this automated machine learning, was not so easy using their platform. So we use this open source package, and from non-plant area into the plants, and then we have been combining this IoT data, different sensors, which I was showing, RGB, multispectral, and thermal for now, and how the weather data, combining all that fusion of that data and applying that AutoML, how we can predict or classify the plants. And it has been really fast with rapid development, quite accurate, state-of-the-art machine learning algorithm, and quite reliable across different crop types and different crop species and multiple environment. We have been testing that, and quite flexible, that a different type of data input can be handled, the approach we have been applying. So we tested that first for the wheat lodging assessment. This is the field trial at the late stage, and where we had significant lodging in some of the sensitive genotypes. And using that machine learning algorithm, first we were able to differentiate yes and no for the lodging, and then we were able to give the lodging score at a scale of 1 to 100. And we recently published this one, and again, here it is showing that how the AutoML work compared with different other model and other approach in machine learning. And cool thing with AutoML was that it was significantly fast in terms of analytics. So you can really do the batch process, and accuracy was quite similar to other approach. And nice thing is that you, in those other approach and network, you have to have like a user input all the time needed, but this AutoML, we have find that okay, it work autonomously once you have all the input parameters added there. So that was the classification yes and no accuracy, and this is the lodging score compared to all the other network and parameters, and it gave really good correlation for this, giving the score from 1 to 100 for different genotypes that have been lodged in that way. And again, this was more than 40 times faster, and deep learning network only accept RGB images, but the AutoML approach can accept multi-channel images like multispectral and hyperspectral quite easily, that we have done quite recently. And again, sort of this is just a sort of an overview of workflow, how that AutoML can work not only for lodging but any other given trait. And we have tested for this wheat field trial I was showing for heading date, maturity, and yield to a really good accuracy for the estimation of wheat yield. And then next phase, we are trying to get how this hybrid data layer using sensor, weather, and soil, and multi-year, multi-location trial, we can use to fuse all the data and applying that AutoML approach to estimate heading date, maturity date, which is phenology. This is quite laborious for us because you have to take the timing, exact timing. And then we are going to have those rust resistance disease resistance for wheat, but also for other crop species as well. And finally, aim will be to do the grain yield prediction. In the last slide, I'm showing here that we are taking that phenotyping to the next level. Most of you might have heard, and some of you might have used the LiDAR sensor, but LiDAR gives the 3D output, 3D point cloud, and that 3D point cloud, when you want to take it for your data estimation, you have to use the proprietary software for the company which you receive the LiDAR. And most of the LiDAR, they are quite expensive, ranging from like 20, 30 thousands to over 100,000 from ground vehicle to the UAV system. But we have recently developed this our own LiDAR system, and it doesn't give the it can give 3D point cloud, but it automatically give you the Excel spreadsheet where you have number for your given plant or of your given plot, and it gives average height, median height, and bio volume. And nice thing is that you don't have to download the data and process it. It has embedded Raspberry Pi computer, which does the onboard real-time analysis computation. And then it works like an IoT as well. As soon as it comes into the Wi-Fi range, it starts to upload the data immediately to the cloud or to your defined destination. And those capabilities number 2 and 3, real-time data analysis and automatic upload, I think that's the really novel. None of the commercial companies which provide LiDAR or any other sensor has been able to do that. So that's sort of our entrepreneurial side of thing, which we are going, and we are starting to make it like on a commercial level production, but mostly it will be used for the research for the projects which we are handling and our collaborators, and then we will go to the next phase to provide it to someone else who is interested. And we have used this for wheat and forage crop and canola. It's worked really nicely to estimate, to give you the bio volume of the not only the plant but also the whole field plot. And then in this final slide, showing our sort of vision, how in that precision breeding scenario, we are combining this genomic data with advanced phenotyping data I have shown, and this weather data and soil data, which is critical if you want to analyze the multi-location, multi-environment. And we have a nice facility for the grain quality, and how all this information and data can be analyzed centrally, and using this machine learning or genomic selection or any other prediction model, how we can sort of get to the plant breeding traits more effectively and more efficiently. So for this precise breeding, precision breeding. With that, I would like to thank my team, Phenomics Group, and all the other collaborators. Many thanks, and sorry for the initial interruption, but I hope that you got the message, what we wanted to give. Thank you very much.
Thank you, Dr. Suryakant. You have made us proud. I'm very happy to hear you after you left for Australia. And I have a set of three queries. One is that how correct is phenotyping? It means that when you go for hyperspectral images, what could be the possibilities of artifacts because of microclimates, because of shade effects, because of dew, because of fog? What could be the artifacts and how they can be removed by the cloud? Second is, if I have an experiment, laid out experiment on critical wheat and durum wheat, and they are genomically diverse. Wheat is AABBRR, durum is AABB, and triticale is AABBDRR. Okay.
Yep.
If I make their hybrids,
Yep.
wheat and triticale hybrids, I
Yep.
will get a combination of AABBDR. can I diagnose through artificial intelligence or spectral image analysis that this is going to be a translocation? Okay.
Yep.
Can this be pointed out? Another thing, third thing is that we have a lot of differences in gluten strength. And based on gluten strength, the use of wheat is decided. It's
Yep.
good for chapati, it's good for bread, it's good for biscuit.
Yep.
So can such minute level differences of grain quality based on gluten strength can also be identified?
Yeah, those are really good question, and I can say that your knowledge and wisdom is getting sharper and sharper.
Yes.
Yeah, coming to your first question about the artifacts,
Yeah.
for hyperspectral and multispectral, that's not a big issue for us
Yeah.
because
Yeah.
it has embedded irradiance sensor sitting on the top of the camera or sensor.
Mm-hmm.
So
Mm-hmm.
that irradiance sensor automatically adjusts the ambient light condition, whether it is cloudy, partially cloudy, or sunny. So it
Mm-hmm.
automatically adjusts in real time the visible scene, what it is seeing. So it removes that artifact. But definitely for those high resolution RGB cameras,
Mm-hmm.
you have to take those consideration in view that okay, you are not looking the shade or you're not looking the dew or mist or whatever. So there are like particular time points which is recommended to fly, for example in early morning.
Mm-hmm.
And for thermal camera is the same thing. The recommended window of flying is that between 10 to 2 o'clock. So that is sort of like quite stable temperature condition. But there are different challenges and different issues with different sensor type. But I think most of them is been now common knowledge available in publication or word from mouth. So that information sort of really you want to utilize and apply when you are taking those observations from a given
Right.
camera. Now, coming to your second question, I think that will be really marvelous to do the hybrid estimation to the hyperspectral. I'm sure no one has done that, but in this advanced era of this science and technology, I would say things can be done.
Right.
We have not done it. But your third question was for the gluten content. Yes, definitely. In our institute, we have grain quality, really fantastic lab that is sort of like top lab in Australia. They work on 30 different parameters for wheat, not only the gluten, but also the dough strength, dough stability, dough color. So a lot of those parameters, they have recently acquired a hyperspectral system. Earlier they were using NIR IFOS system, but now they have this Specin company system for the hyperspectral, and I have seen their initial result that it is nicely picking those valuable data set in a non-destructive and high throughput manner in the grains.
Very good. Very good. Thank you. Thank you, Dr. Suryakant. Very
Yeah,
nice.
yeah. No,
Thank you
thanks.
so much, Dr. Suryakant, for your valuable presentation on such a wonderful topic. Now it's turn of Dr. Tino Dornbush. Tino Dornbush is a phenotyping solution expert at Dhiom Lab UK and currently based and operating remotely from Germany and looks after most of the implementation strategies of enterprise-oriented custom solutions and pipelines for interdisciplinary plant science and life science-related projects. Since 2003, both in academia and industry in European Union, his expertise covers technical aspects on sensors and automation technology, data analysis, visualization, software development, and communication of scientific technical content to experts as well as non-experts. He have been working on image analytics covering satellite, field, greenhouse, and lab applications. These skills have been crucial to successfully work in interdisciplinary R&D projects where he has interfaced with customers, scientists, technical engineers, computer scientists, and business stakeholders. I would like to invite Dr. Tino Donbush to present his view on using artificial eyes and intelligence to assess plant disease, perspectives, and challenges. Welcome, sir.
Oh yeah, thank you for your introduction. It sounds from your words that I'm a genius. That actually I'm not, but hopefully audio is coming through. My presentation is advancing. Yeah, can I confirm this?
Yes, sir.
Okay, good. Thank you. So, namaste.
Namaste.
Yeah. So today I want to give you my viewpoint on how to use artificial eyes, speaking of sensors and artificial intelligence, to assess plant diseases on a very general level, a very general introduction. My presentation still visible? Oh yeah, okay. So on a very general level. All right, so let's see. How can I now move on here? All right. So I think we have a common agreement that climate change is threatening ecosystems worldwide on this planet. As you might know, that Germany would have been covered by more than 90% of forest without human activity. And still, until now, we have around about 32% of our area in Germany covered with wood. And the climate change is dramatically altering the plant vigor, the tree vigor, and now this is very susceptible to diseases. And it is projected that the forest will decline in area, and it's very difficult for us to cope with that, right? I mean, I could have picked any picture from any region of the world. So we all know that climate change is affecting plant health, plant vigor, and it makes them more susceptible for disease. So it's important to understand this better. Artificial eyes for monitoring biological objects. So Surya already have given a lot of examples on what kind of sensor technology is available. I just want to give you a very general overview. So this is like the light spectrum that goes from ultraviolet light visible until the infrared. And for the different part of the electromagnetic spectrum, we have different sensors that can look into this, right? So our eyes, or the RGB camera, is looking into visible range from 420 to 680 nanometers. There are now also hyperspectral cameras that are able to monitor in the ultraviolet light. Then we have the visible near-infrared light cameras that cover a range from 400 to 1000 nanometers. We have the sphere shortwave infrared cameras that cover 900 to 2500, and we have infrared cameras. Nowadays, also new cameras are developed to cover the range in the medium and the long wave infrared, so bridging this gap. But commercially, they are still in the let's say in the infancy, in the beginning of their work. So those sensors can be mounted on almost any different appliance, right? So we can mount sensors on microscopes. We can have handheld devices. We can mount them to vehicles, to drones, to planes, to satellites, right? And as the sensor to object distance is increasing, the resolution is going down. But at the same time, we can also image a much higher area, right? So that means the proper choice of your sensor and the platform is crucial, very important to monitor plant diseases. Moreover, there are also a lot of automation technologies available. I just pick out some examples, for instance, for the lab, right? So you all probably know that you have different types of microscope: light microscope, confocal, two-photon microscopy, where we can really image on a cellular and subcellular level, right? So we have imaging solutions where you have boxes in which you can subject your samples and you can image them. You can also have boxes with a certain degree of automation where, the camera is moving around, where you can image a larger area. Only a small example of this. We can put these sensors in greenhouses, right? By having two general approaches. We can move the sensor in big boxes over your plots, over your targets, or we can move the plants around in the greenhouse, what Surya was describing, where they all go one by one into imaging chambers and they are being monitored by the artificial eyes or by the sensors. We can have different automation technology for the field, right? So we can again mount the cameras on a big gantry system. We can have them on small robots or on tractors that go over the field. We can mount them to drones, and we can mount them to satellites, right? So the automation technology is there. So here comes now the problem space. We have a large degree of sensor and automation technology available, right? And what also Surya was pointing out, the amount of sensor data is rising. So there are petabytes of data produced every day that need to be processed by all the different sensors, by satellites, by drones, by cameras, etc., etc. And the bottleneck until now, it is the image processing, the feature extraction, and more importantly, the correlation to the biological data that is relevant. And so the question I'm raising here, does AI solve the problem? Hopefully by the end of this talk, I will have you figure out an answer, or, well, you make up your answer. So just go back to a very, very basic detail about how these sensors are working, or our sensor in general. So we always have to keep in mind the four important components in this system. So first of all, we have our light source. In most of the application, it is the sun. And you all know that light is composed of an electromagnetic spectrum, which we can see. So this light would now hit an object of interest. In this case, it is a red apple. It is reflected and then captured by the detector. In this case, it's our eye. And I just try to point out that's like how we perceive the color is the object is, in this case, the apple is reflecting mainly red light. So it's absorbing the blue light and the green light. And so the red light of the spectrum is detected, right? And now the magic happens that I have never understand. So the retina in your eye is transforming this electromagnetic radiation, this energy, into an electrical signal that is ultimately processed by your brain, by the neurons, right? And so the brain sees and say, okay, this is apple. And the apple is red. And I can take a decision where I want to eat it, let's say. And a human takes around about, I would say, 1 year to master this. From the stage you are born until your neurons have been set up to make the association. Okay, this is an apple, this is a color, and also to do the motoric movements to eat it. So the same thing is we can now look at a diseased leaf, right? This is like an example of soybean with rust. So if we're looking at the healthy spot, we see that mainly green light is reflected, right? And if we look at the healthy spot, we see that this shifts towards the red light of the spectrum. So we see color differences in our sample, right? But in order to learn this, to say, okay, first of all, to detect this is a soybean leaf, this is a disease, and this is that disease. It takes a learning and development process of probably more than 20 years to become a phytopathologist that actually knows how to address this, understand the biology of the plant, understand the biology of the pathogen, understand the plant-pathogen interaction, and to see what is that. So obviously, what we want to do is we want to exchange the sensor, our eye, to a camera, and we want to exchange the brain, right, to make the decision-making system to a computer, artificial. We want to make all these things artificial. Okay, so I mean, you probably know all this, but I just wanted to, for the sake of simplicity, explain that what is an RGB image. It's actually for the computer, it is nothing else than a series of numbers. Right? For each pixel, you get the different values, red, green, and blue values. And depending on how these values are composed, or the height of this value, it creates an image that is being displayed on the computer, and you can see it. If we now looking at hyperspectral images, so we increase the amount of layers, the amount of images, or the amount of numbers that we see for each pixel, right? So if we talk about visible near-infrared cameras, each pixel can have up to 1,600 values, and you see all these values create what we call a spectrum. The advantage here is that we can cover the near-infrared range, which human eyes cannot see in this range, and we have a much denser data package that we can extract features from and to take a decision. But again, comes the problem of increase of data and the problem of processing all this, storing and processing all this data, extracting of meaningful information. Okay, so let's take out a very simple example here, right? So the use case, we'll stick to our soybean rust example. The task for the AI would be, okay, determine whether a leaf is infected and shows symptoms. And the second, assess the disease severity. So a human trained pathologist, and even most of you can clearly see, okay, well, this is a very healthy leaf because I don't see any of these lesions here. Here, this is quite thick. You know, we could easily make up a ranking because of our cognition, because of our experiences that we have. But the computer cannot. He just sees numbers. So we have to train and we have to teach him. So there are two general approaches to classify images for the computer. So we can do a pixel-based classification. So pixel by pixel, we look at it and say, this is a disease, this is healthy, and God knows what, right? So we transform our initial image into a color-coded image, right? Where each color has a specific meaning, right? And we can analyze then the amount of pixel of red pixel, of green pixels, of blue pixels, and say, we can derive a leaf area. We can then say, okay, this is how healthy it is. You know, how much % of healthy, disease, chlorotic, non-relevant pixels. And we can use this information now to classify our leaves, grouping. The second approach is that we take the entire image. We're not going pixel by pixel basis. We take the entire image and we classify in a very simple stage, healthy or disease. Of course, you can define a much larger scale of or ranking of diseases, right? But for the sake of simplicity, just say, you know, healthy or disease. So for the machine learning itself, there are also two main approaches what have been called. First one is what people denote as machine learning. So we have actually our input image. Then the user defines a set of features, characteristics, for instance, color features, shape features, etc. That are then used to train your classifier. Or you use a classifier, right, to say, okay, if the pixel value is bigger than red, if the red value is bigger than whatever 50, then it is healthy. If the red value is smaller than 50, it is disease. So a very simple classification that are available, right? It can be neural network, it can be simple regression, you know, it can be decision trees, God knows what. So the main difference for the deep between what people call machine learning and deep learning is that the entire model itself, the feature extraction and the classification is everything in one, right? So the deep learning decides which features to take, you know, how to fold all those images, you know, to get the information. And the model essentially is a black box, right? You train it, and, you know, not even the computer specialist probably will really understand, because of all these different connections between the layers, between this artificial neurons. And in the end, honestly, who cares, right? It is a model. It produces the output that you want. So you could always argue, why do I care? But keep in mind, this is like a black box. Okay, so now here comes the problem. So how do we actually now train the computer or the model, right? So he just gets a set of numbers, right? An array, red, green, blue value. How do I know it? So we have to define, first of all, our different disease stages or our different groups, right? We have the background healthy symptoms. So this information needs to come from the domain expert, from the pathologist. Okay. So what you have to do is you have to take your software of choice, Photoshop, GIMP, whatever tools, and you have to create an annotation layer where you manually have to paint the pixel with the corresponding color and say this pixel is healthy, right? This pixel is disease, you see, and this is a background. So you really have to give them a large variety of input data set for the training. So this is like, again, the images annotation layer. This we're going then for the model training. So this is now how it would look like in the end if you have trained your model well, right? So you have these are the input images on the bottom, and on the top you see color coded for each pixel what the system thinks it is, the pixel is, right? And the corresponding to the defined classes. And now you can use this information, as I tried to explain you before, like the fraction of the different pixels in order to now group and quantitatively analyze your disease severity. Okay. So how does it now work for the model training for the deep learning approach? So you can imagine that this procedure is quite tedious. I can tell you that. I've done it myself multiple times. So annotating all these things correctly, it takes a lot of time. And you also have to annotate, depending on the biological variability, 10,000s or 20,000s or even more than that of pixels, right? So the training for the deep learning approach, where we use the entire image, is much easier. Right, so we have to group our images into the desired classes and to give them a tag, or okay, this is healthy, this is sick, this sick, healthy, sick, sick. So, right, but for this training data set, we also need much more than 1,000 images to successfully train the neural network or the convolutional neural network. So it's also a lot of images that you need to stuff in there. And then, of course, we need to have the annotation of the ground truth data, right? So tell them sick, healthy, or whatever tag you want to give your leaf, and you have to do the model training. So this approach also comes with a certain drawback here. So the images that you are using, they all need to be of the same dimension, height and width. So you cannot just take a 1 megabyte pixel 1000 by 1000 image, or you compare it with a 2000 by 2000 pixel image. That doesn't work. So all they need to be at the same size. The computational resources required are quite high, depending, of course, on image size and so on and so forth, right? But compared to classical regression or classical classifiers, that requires much more computational resources. And the model, as I said before, is simply a black box, right? You have no idea what's going on inside there. So, does solve AI now the problem of analyzing large image data sets to assess plant diseases? Okay. Right. So the problem is that we still have to overcome a lot of problems here, or challenges, right? So there is the biological variability, right? First of all. Right, so coming back to this example, is a very simple, very good example, because we have flattened out the leaves. We always have the perfect angle to monitor it, right? But the reality is different, right? Leaves are moving in all directions. They overlap, right? We have variability from the different varieties, right? Soybean varieties, for instance, we have different strains of diseases that interact. So, strain A might be reacting differently with cultivar A than strain B with cultivar C, etc. So we have quite a lot of variability that comes from all different sources, genetic variability, and so on and so forth. The second thing is sensor calibration, especially if you work under non-uniform conditions, right? So outdoor. So Julia was pointing out that, of course, you have to, if you do drone imaging or you do satellite imaging, you always focus during the midday when the sun angle is the highest, so to get reproducible conditions. And you constantly have to normalize your data, right, to get a good and comparable data. Then keep in mind, this is a very important aspect here, that we have the physical constraint, right? So light does not penetrate very deep into plant tissues, right? Depending on the energy level, right? So UV light is like 10 nanometers deep. And the more wavelengths, the higher the wavelengths is, the deeper it penetrates into the tissue. But keep in mind, if you want to see it, the photon that comes into the leaf also need to get out to the sensor. So you actually have to halve the penetration depth. So that comes to a problem if you want, for instance, monitor diseases that are spreading inside the leaf, right, in the leaf veins or something like that. So you cannot simply not see them because, the light doesn't come to it and cannot be reflected. Another constraint is that the plant pigments are the predominant absorbers here. So they are dominating all the spectral signatures. So that's why also when you do whatever confocal or to photon microscopy, so most of the time you are clearing all these pigments out of your sample, right? If not, you would not see your spectral signal, right, of your fluorescence protein, for instance. So very important. It also means when you want to monitor diseases, most of the time what you do is that you monitor the changes of your different plant pigments. So you visual symptoms that you monitor and the indirect effect of the plant pathogen interaction. So implications are disease detection are highly constrained to plant surfaces, so on the outer side of the plant, right? And indirect disease detection via the degradation of leaf pigments. Okay, so again, now what is very important, understanding the physical and the biological constraint, but also the choice of the right sensor mount and the imaging distance, right? It clearly depends on your question, right? We had it before. We can go to the laboratory where we can do electron microscopy, stereo microscope. We can move up the scale to optical systems where we image at a very close distance at a relatively high resolution. And the more we go away from the sample, the higher the imaging area is, but the lower the resolution to see all these effects that we want to see. Okay. So does AI now solve the problem of analyzing large data sets to assess plant diseases? I would say my opinion is currently now, no, but it helps a lot. You still have to have an understanding about your biological object. You have to have an understanding on how the sensor works. But the good thing is teaching AI with your intelligence will solve the problem. So we need a generation of young people that are now able to learn all these things, and we have to learn them by heart in order to train the computers, right? So it would still take some time, but I'm quite sure that we or you are going to manage this. So thank you. That is my final conclusion at this stage, and happy to take any questions now or later, or depending on, what's the policy here. Thank you.
Thank you, Dr. Don Bush. You have clarified my first question about artifacts, and that is what I wanted to know, that what are irritants in our image analysis. and genotypic variability, genomic variability, environmental variability. You have all cited wonderfully, and that has answered my question. My question is, at what stage we can identify disease? I give you an example, and then you give me an answer.
Yep.
In India, North India, the rust pustules, they come from southern India. They from Nilgiri hills, along with the clouds, monsoon rains, and they fall on the plants growing in northern India, wheat plants.
Yeah.
Okay, and nobody knows, nobody see them that whether they have come or not. The people see only when the plant becomes diseased. Okay,
Yeah.
and it takes a while. It takes a while. When the spore is put on the plant, it will germinate, grow, and the hysteria of fungal hysteria will grow within the leaf tissues. It will become mycelium, and mycelium will give rise to secondary spores, tertiary spores, primary spores. Okay, can our systems intelligently work out that the spores have fallen on the leaves and here is the time to have control measures in place so that no disease occurs? Or the very beginning hysteria have been developed or mycelium has been developed and we need to arrest it. That is second stage. Primary sporulation has occurred and we have identified spores. They are still low in numbers and the many plants are not infected. We can, at that stage, spray the plants. Can that accuracy be achieved by artificial intelligence, hyperspectral analysis?
Yeah, I mean, from your understanding, from my understanding, it's a fungal disease, right? So
Yeah,
the
fungal
spores
disease.
falling on the leaf, they germinate, they affect they grow inside the plant and the leaf veins
Yeah.
and the okay,
Yeah.
understood. Yeah, so first of all, you would need to have a very high resolution, right? The spores are very small and the fungal structures are also very small. So
Yes.
don't try to go do you satellite imagery or drone imagery to detect it at this stage. Then the next problem is that you probably the spores are too small to detect them. At least to my this is what my take on it. Even if you would take leaf samples and really image them at a very high resolution, so it would be possible if you knew the spectral signature of the spores itself. But again, you would need to have a very high resolution, and I'm not sure whether this will work out. Another thing that you have to try to tweak the system the way that or let's say you have to educate your plant to tell you, okay, there's something. So you would need to have introduce some reporting mechanisms, right? So there have been studies showing that when plant defense is activated, that luciferin, like a fluorescent a fluorescent system, is being active in the dark. So that could be a way to, for instance, also to monitor this. But again, then you would need to have highly standardized conditions in the laboratory.
Very good, very good.
So
Because
on the field scale, sorry.
Yeah, I was also thinking that the pigmentation changes can be trapped easily than the spore itself or the disease itself, and that those changes can give us enough hints that the plant is likely to be diseased. And that you have already said that the fluorescence coming out of the plants can be sensed very easily. That is being done, and that may be one reason. Yeah.
That reminds me also, there are some systems or some approaches that try to filter air and try to detect spores of different kinds using machine vision.
We use spore traps.
So that
but
is
most of the time they are washed. Most of the time they are washed by rains.
Again, this is one of the key messages that I wanted to convey. The experimental design is crucial here. The sensors and the computers are honestly stupid. They're just converting light into digital numbers and digital numbers into something that you train them to do with. So very
Yes.
important is to have the understanding about the biology in order to deploy the sensors the best way you can use it. So this is core importance of all these phenotyping approaches.
Yeah, sometimes there is a wax deposition on the leaves. So we need to standardize by sensors, maybe for each variety.
Yeah, oh yeah,
Because
that is another problem. Yeah, so once you have made up or you trained your model and your sensor combination for variety A, it doesn't work for variety B or
yeah,
for whatever climatic condition C. You know,
yes,
this is another problem here. That's why
yes,
I say AI does not solve the entire problem space, but it
very
helps.
good.
It helps.
Very good. Your lecture has been very informative. Thank you.
Yep.
Both lectures have complemented each other. Suryakant and your lecture, the eye-opening. Okay, thank you.
Thank you so much, Dr. Donbush, for such a presentation on important topic. Now it's turn of Dr. Sudhir Kumar. Dr. Sudhir Kumar presently working as scientist in Indian Agricultural Research Institute. He obtained his PhD in plant psychology from IARI New Delhi. He has authored many research articles in national, international journals of reputed. Besides these, he has published training manuals, technical bulletins, popular articles, and books. He has served Indian Institute for Farming System Research, Modipuram, and Indian Institute of Agricultural Research, New Delhi. His main area of work is abiotic stress, plant phenotyping, cropping system management, image-based phenotyping, and sensor-based plant trait dissection. Now I invite Dr. Sudhir Kumar to present his view. Please, sir.
Okay, very good afternoon, and it's my pleasure that the organizer has given me this opportunity to explain briefly about my work. Prior to that one, because the last question comes for the leaf rust, so I have few images, so it will be a good idea to show that, and then some insight of the information can. I hope my screen is visible to you people.
Yes, sir.
So these are the few images if Tino can see, and I hope they can get more insight how the leaf rust look like in the initial stage, and once it shows severity, how the leaf get changed. So these are the initial stages where the leaf rust just start, and in the last where the leaf look like there all the photosynthetic activity goes off. So these are the things I want to intend to show, but now I think I will go forward with the whatever being assigned to me. The thing currently I want to share, just a moment, that screen contain, I hope it's the one. Are you able to see my screen now?
No, sir.
Okay. Okay, I expect that. Yes, just a moment, just a moment. Allow. Okay, now I hope it's you could able to see my screen now.
No, sir. No. It's not visible.
Not visible. Okay, okay, please, please. Let me try one more time. Now it's okay.
Yes, sir.
I am audible, so both things are working nice. And but the strange part is that two advanced persons come prior to me, and they explain the things very nicely about the plant phenotyping, and now I'm being asked to explain basics about plant phenotyping. But anyway, whatever information I have I will show, and whatever I could share here I will. So everybody is working for improvement of crop for sure, and to see the things from a distance is somewhat different, and to know more about them is somewhat different. So the most important aspect in this whole plant world is how the plant actually appear, what we could see in terms of plants. And there are few basic informations we know. Whatever the old traits which can be appear for a plant which has its own genotypic base, and based on the environment interaction how it can look like, there are several uh, name has been given. For instance, we use phenomics, where we use phenomes like genome and genomics, but here phenes
Screen has been freezed.
include all.
Sir, your screen is freezed.
My screen is freezed.
Yes, sir.
Now it's okay.
No,
No, sir.
Okay. God, what's wrong with my screen?
sir, we are not able to see your slides.
Okay, just a moment. Let me go out and then.
नहीं नहीं उसी को Okay, okay.
Okay. Now it's okay.
No, sir.
Yeah, I'm just seeing, sir.
Yeah.
It's fine now.
No, sir,
No,
it's not
sir.
fine again.
Ah, my goodness, what's wrong with it?
शेयरिंग के लिए विंडो सिलेक्ट करनी
You are
पड़ेगी.
in show. Everyone can see you here. Shall I stop my
Sir,
video?
you are seeing you are sharing your entire screen. Kindly share
Yeah.
one window only. That is of PPT.
Yeah, I am sharing that only.
Please
Just a
select
moment.
PPT only.
Okay. Let me try. Share screen. It says that presentation 2. No entire screen. Okay. Let me try that.
Don't say entire screen.
And then it's okay now.
Yeah,
Yes,
now
sir.
it's okay, sir. Now
Okay.
please continue.
Okay.
Okay, fine. So I was here. So
Yeah,
we
now it's
use
okay.
if we use a few terms like phenomics, and the things what we could see through eyes, they can be keep in eyes, but when we have to analyze the information in terms of plant, we have large number of genotypes. So those information we cannot keep in our eyes. And the most important aspect what we could see in terms of plants is based on is based on genotype and environment. Even this environment play a key role. So there are ways, as I am focusing on basics parts, there are ways to get information. Either the plant has been sent to the sensors, or sensors, a non-invasive sensors can be used for data acquisition. And the third part is that all the data which is obtained rather is being used for phenotype the traits. As it's explained by Dr. Suryakant, why we are focusing more on this HTP, high throughput phenotyping, because of the large labor and investment and accuracy. But HTP, or the high throughput phenotyping, still has few bottlenecks, and out of that one is the one hardware is the one aspect. Then we have data management, because the kind of sensors we have, they acquire large number of data, huge data. Then the third aspect is that whatever information we are obtaining, that's also not only in terms of images, but that's also time series data over the time, say 10 days, 20 days, how the plants are behaving. And that's not only one genotype, we are focusing of large number of genotypes. And then the another aspect like analysis, the complex, hard to make, turnkey. And whatever we have done up to this stage, HTP, in the last, how we actually publish this data, sharing the codes, ultimately to get all those information, high throughput aspect is one, but our main aim is always like to get best out of the available information. So going further, few information still has to be added. There are two ways. One way where we can exclude out environment part, where we are having control environment part, where we grow the plants and give the same kind of conditions to all the plants, and let's see what are their genetic potential is there, and that can be captured through the camera, where each plant is being called to the sensor and the image being taken. The second aspect is that in the field condition, how the actually plant is growing. So in that field condition, one aspect is the mobile platform where we can have the plant has been grown and we have a frame on which camera can move. And the second aspect lies the drone. Drone is the one aspect which can be say attached with large number of sensors, and those sensors can get actual information what is happening inside the field itself. Then the whole exercise, whatever information we are getting, is for this screen only. It may be above ground part, how the leaves are behaving, how the roots are behaving, what are the actual aspects, because up to this time we are focusing only on breeding. But now the time has come, we are not going for empirical breeding now. We can go to the field, find out a few good genotypes, and then go ahead with the, say, all in the trial. But the thing is that now we need to go to specific trait based breeding, and these traits can be obtained only after having a good phenotypic data. And once we have that phenotypic data, that only we can go for directional breeding. And for this directional breeding, some kind of terms, water use efficiency, hardly we hear for improvement in the crops, except a few cases from the Reynolds who did say from Australia, and then harvest index. These are the secondary traits which we are get from this high throughput phenotypic platform. And out of these, a few important more like transpiration efficiency, water uptake, these kind of information can be obtained in real time. And as I told in the few earlier slides, we can get this information on time scale. So once we have this information on time scale, then stage wise and genotype wise, when we bring all those things together, we can find some interesting traits, and those traits can be put into the breeding pipelines to improve the crops furthermore. Then the second aspect, whatever we do, we require the backup, how these light, camera and action can come into the one frame, and then they can see these plants at different stages with their different spectrums. Based on that, when some may see in the visual range, some VNIR range is being explained by Tino, how these ranges look like for hyperspectral SWARE and say RGB or NIR, but still each spectrum has its own peculiarities. Once that specific peculiarity has been obtained, that peculiarity can be used to translate into the traits. And once we have those traits, we can go forward with more improvement in the crops. Then these are the few images which help us to know how actually these whole plant phenotyping process goes on. For instance, if I start with NIR, it gives idea how the plant water status is there. Then comes the RGB, what we could see through eyes. It's same can become by the same sensors, and it gives idea how our canopy is spread, how much green it's, and how many say up to this time, as is been told earlier by 2-3 presentation, that artificial intelligence come into the picture, that we can know how many tillers are there. So those kind of information can be obtained by these RGB sensors. Then becomes the root imaging, key aspect. Though we cannot go deep into the soil, but still some efforts have been put to bring out information about the roots. Then come infrared, which gives indirectly the plant temperature, though it's plant temperature, but it's also give idea how my roots are behaving. Are they able to capture the whole amount of water from the soil and can translocate to the leaf? Are they efficient enough? So that kind of information can be obtained from infrared. Then comes the crop reporter, as told by Tino. The fluorescence is the one which can give information how effectively my leaf are working prior to that infection. For instance, my leaves are healthy if I take those informations on day one, and after the say reaching of spores, those two images can show that some kind of infection has started. Those cannot be seen through eyes, but these sensors can give us idea, yeah, plant is under stress. Then these are the environmental conditions where we move the plant, because inside the greenhouse we have some kind of position effect. So if we keep moving these plants, the same plant cannot be stick to one situation, so it can be all kind of environmental conditions. So those factors can be taken out. And beside these imaging sensors, two more sensors of key importance. These called hyperspectral sensor. Range starts so say 400 to 2500. That can be split out into two. One is SWARE and one is VNIR. VNIR range from 400 to 1000. And in case of SWARE, that can range start from 900 to 2500, and these can be used to get more information like biochemical parameters. Maybe as Dr. Suryakant told, the chlorophyll can be estimated, and some information has been obtained that NPK kind of work has been started from UK, and so this large information can be obtained by these two sensors when we go for plant phenotyping. I hope from this screen what I want to make out of this, how the plant actually start from the day one, how the plant is growing, what are the biochemical changes take place, and at what stage the plant is reached. So these kind of information can be obtained by these sensors as whole. So our focus always lies phenotype. Phenotype, the words being a plant physiologist, being a artificial intelligence guy, CNN, everybody is interesting to get information, how the height is changing, how the growth rate is changing, how number tillers are there from the images, how we can get those informations. And still now, because these are the initial traits, the ultimate objective of each experiment in case of plant field lies how much yield we can get or how much yield improvement we can get. So these kind of information can still be obtained by this kind of sensors. But the real fact lies how much efforts we put in training our computer, maybe whatever the machine based algorithm we use. So that can be, once we train our machine, then it can further use those kind of trained information to get actual information from the images what we could have from our experiments. Then these are the few traits, and this image is the one which shows that the plant activity, how it's changing. These are the actually fluorescence based images, and these are the RGB images, how my plant look like. So these kind of information. Then this screen gives us idea. For instance, if we are taking images at each stage, these are the two separate experiments, where the one where up to that stage, say for instance, we allow these two ports allowed to grow in the same conditions, but from here onwards we put some kind of regulation on watering, and you could see the effect, how fast senescence take place. And if we use the artificial intelligence based information here, how many leaves are changing from yellow, and how many leaves are green here. So these kind of information can help us to find out from the large number of genotypes, though these are only two ports. But when we have large ports, large genotypes, large accession numbers, to get information from that one, it's required only machine based port, because our eyes can at once see 2 3 maybe 4, but to see large number of and with quantified data required more and more accuracy in that one. Machine can help us a lot. Then these are the flow chart or the pathway which can be used, and we mostly focus up to this stage. Everybody around the world has reached up to this stage, but the real focus lies here, how the classification of all the information we have obtained can go forward with this one. Then these are the simple process which shows that how our interest for say image, which part of our interest are, and what kind of information we can get, and what the available softwares, maybe some are paid, some may be free. Based on that we can get our features of interest. Then these are the few images which we separate out from say facility we have here in Ahira, and it's the one interested one, but still with some kind of limitations that below ground study can be done, but it requires some kind of efforts, and we have to use special kind of ports. Then these are the information can be obtained, how my root architecture is actually spread, and it also gives idea based on the NIR sensor how the water is actually spread. So these are the second aspect. Then even though if we have a very sophisticated facility, fine, but still if we don't have, the real aspect is that if we can keep some kind of uniform background and a RGB camera, it still can be useful to get information about the plant at each stage. So these kind of background say instance, and we can use to get more information, even though if we don't have these kind of facilities. So these are the free software like images available, and we can get area and height at instantly out of these images. Then we have that, what about that high throughput method for yield? Because as I told, that it's not the only part that we should focus on traits, but the real part is that are we able to predict the yield? And it has been done to taken out years from the wheat, and based on the number of years it was applying a thresholding was applied, we know the number of years, and some manual intervention, so we know that how much seeds are there in few, say for a few years we train those, and based on that we can predict yield. And this was published in Plant Methods. Then one aspect, seed is second aspect, how the seed size change, seed color change, seed yield change. So all those kind of information can be obtained in a simple way. Scanners can be used to get information about this. Then uh, still the real question lies, what kind of software we have? Some softwares are paid ones, some are free, so based on the pocket and the money availability we can go forward, but still we can get the information for the seeds in a good way that can be used for predict yield or the seed quality. Then say area, length, all those features can be obtained by these kind of softwares. Still I was focusing up to this time control environment, but if we are focusing to go outside in the field, drone can help. And these kind of information as told by Dr. Suryakant, still we have drone kind of facility, and they can give idea how that drone, because mostly we are working on say nutrient and water use efficiency. So this kind of information can be obtained, but still we are in the preliminary phase, and still more work has to be done in this field. So the take home message, yes, few information from RGB camera, even if we don't have expensive one like hyperspectral camera, still greenness and sunny sense, and even economic part can be taken out from the images, and these can be used for our experiments. So these are the team. I like to thank all my team members who helped me and who guide me to reach up to this stage. I will be happy to take few questions in this regard. Thank you very much.
Hello. Hello, Dr. Sudhir Kumar. Thank you very much
Sir,
for your nice presentation.
sir.
I have one or two questions to you. You see that in case of wheat, we both are working on wheat. In case of wheat, canopy temperature is one of the best criteria to identify the vitality of plant, whether it's photosynthesizing or whether it is under stress. Canopy temperature. It can come the same through two ways. It can come the same through two ways. One is by transpirational cooling, and another is by reflection back of the light. Okay,
True,
in
sir.
one genotype, the heat wave, the light waves are absorbed more, and the transpirational cooling balance it out. And another genotype, the light waves, they accumulate, they get reflected back to the environment so that they don't raise the temperature so nicely. Can these differences, minor differences, be isolated by your hyperspectral images?
Sir, if I put a point that we work on the wheat with the wax content, and we found that by even RGB image, we could find out those genotypes which have wax layering, and they have more cooling. And from hyperspectral, though these information can be done, but the real question lies, we have to match those information from the wet lab also. That if for instance my hyperspectral signature showing that some kind of x is there, I need to make that information from the lab also, that how much x is there, and then to see that how much cooling it's affecting. So that aspect like, but still from RGB image we could able to do, but for hyper spectrum images we are working for that one, sir.
Yeah, I tell you a practical problem. When we make F2 population, it segregates. And we have to identify individual plant having all the good traits. So if I have a climate ecology of Rajasthan, I don't need a genotype which will waste so much of water on transpiration. I need a genotype which reflects back lot of the energy or have lot of osmolyte concentration to remain the cellular turgidity in place. Okay, so can this on F2 population to identify individual plants? Then it will help breeders.
Sir, sure. It can be, but the real input is that we have to have a team which focus on both phenotyping aspect and second in the lab, so that information with the lab can be matched, and then we can go forward. Yes, I can say these sensors have the capability to segregate out those kind of things, because hyperspectral, I read 2-3 papers that proline estimation can be done.
Very
So,
good. Thank you.
sir.
Thank you. Thank you very much for clarification. Nice.
Thank you so much, sir, for your talk about such a wonderful topic. In continuation, now I want to introduce Professor P.K. Ghosh. Dr. P.K. Ghosh is PhD, topped in MSc with gold medal from Govind Ballabh Pant University of Agriculture and Technology, Pantnagar. He started his career as scientist in Indian Council of Agricultural Research in April 1993, completed 28 years services in which 11 years as a research management position and 14 years as professor and principal scientist. Presently working as founder, director, and vice chancellor, ICAR, National Institute of Biotic Stress Management, Raipur, Chhattisgarh, India. Earlier, he demonstrated leadership as a national coordinator, National Agricultural Higher Education Project, Bird Bank Edit Project at ICAR, New Delhi from 2017 to 2020. Director, Indian Grassland and Fodder Research Institute, Jhansi, and Head Crop Production Division, Indian Institute of Pulses Research. His pioneering works on climate change, carbon sequestration, resource use efficiency, conservation agriculture, crop diversification, and soil water conservation are highly recognized. Now I invite Professor P.K. Ghosh to present his view about management of biotechnology stresses, use of artificial intelligence. Welcome, sir.
Stop sharing. My slide is visible, madam? Yes.
Sir, your slides are not visible. Actually, you have shared entire screen. Kindly share one window only.
Yes. Now it has come perhaps.
Yes, sir. Yes,
Yes,
sir. Now
sir.
it's good.
Okay, thank you very much to the organizer for inviting me. As madam mentioned that basically I am a natural resource management scientist, now working as a director cum vice chancellor of Biotic Stress Management Institute. Now, as the topic assigned to me, before coming to the actual topic, I just highlight how this biotic stress management is issues, challenges are affecting, and what will be the issues and challenges, and what should be the way forward. As we know that this is biotic stress, it causes huge economic loss to the crops and also the post-harvest crops also, and leading to 18% of the crop loss, and in terms of the monetary value it is towards 60,000 crore. Some estimate also claim that it is more than 60,000 crore, that is 1,40,000 crores. So there is some variation, but in real picture it is a serious issue, the crop losses. Now coming to the major issues, yes, there is a invasive insect pest and pathogens are a serious concern in our, not in whole Asian countries, and in particular in our India, that many new alliance and transboundary pests, particularly the fall army worm of maize and tomato leaf miner is a serious concern. Then climate change impact on biotic stress is another serious concern. Pesticide induced resistance, this is, I think it is still a researchable issue, and I think very little work has been done in our country. And then ultimately the topics of today is early detection and diagnostics of the disease and pest. So this is a very important topic you have chosen. Now coming to, as I mentioned, that climate change and its impact on biotic stress, I think it is hardly we have worked on that, but there is a very, great scope to work on that, how the climate change are changing the biology insect pest and pathogen, and ultimately it increase the distribution and expansion of their population in various locations, enhance the survivability and adaptability, outbreak, and this host shift is also very important aspect to be looked into. Then emerging of new biotypes and race and strains. Now, as I mentioned, that pesticide-induced resistance is another important aspect. So far that I think as per our estimate that 954 pest species has developed resistance against the different pesticide.
Excuse me, sir.
Yes.
Sorry for interruption, sir, but your slide is not moving.
Not moving.
Yes, sir.
Okay, but I could see that it is as per this movement I am going for.
Only
so
first slide is visible.
Stop sharing, please. Okay. Stop sharing. Now it is okay.
Yes, sir.
Now it is visible now.
Yes, sir. Now it's okay.
Okay. I think I covered this slide. Now you see the serious threats of invasive pest and disease. These are the disease and pest and against each crop. The India is facing a serious problem with these invasive pest. And also there is a potential threats of 2 to 3 disease, that is wheat, blast and UG19 races. I think we are aware of it, that it is now is prevalent in Bangladesh and this Iran, that wheat blast, and then there is the potential threats for soybean, that is downy mildew. We understand that biological content, why it is not well adapted. There are so many issues. Consistency in the performance is not there, particularly for delivery system, field efficiency, then good shelf life, and then ultimately farmers acceptance is very low. So we have to take care all these aspects when we are going for large scale adaptation by the farmers. This is some of the issues we have to take care. Now, some of the work on this biotechnological stresses has been carried out in India, particularly in my institute. A very good understanding that if a project is running, that is understanding molecular mechanism for virus, we know that this yellow mosaic virus of bean is transmitted by white fly. But this virus is not affecting the tomato plant and potato plant, and this is and tomato plant, and this is vice versa. To understand the why it is so, there is a experiment where to identify this host and pathogen relationship and non-host and pathogen relationship. So my scientist has identified the virus replication, where it is shown that the mechanism and where we can identify the why it is so. Now, as I mentioned, that identified miRNA technique to how it regulate the pathogen population in chickpea pathology, that is a fusarium against this fusarium oxyporum in chickpea. So this is another technique.
Excuse me, sir.
Yes. Again, it is not.
No, sir. Slides are not moving again.
Dr. Mumantham को बुलाओ ना भाई. Dr. Mumantham, when director is presenting, you should come over here. अब देखो non-stop हो रहा है. They are unable to see. देखो कुछ problem है. I could see, but they are unable to see the slide. It is not moving, as they mentioned. It is not moving.
It's moving here. The slides are not moving.
Not moving. When I just am shifting, it is coming that stop screening. Okay, sir. अब देखो एक बार.
Sir, is it visible, sir? It's moving?
No.
No, sir.
Not visible. Okay, sir. You can continue without the slides. No issue.
Again, we are sharing, sir.
Okay, let me share once again.
So is it visible, sir, our slides?
Yeah, now
Yes, sir.
first slide is visible, sir. It's from
Yeah.
starting. Kindly move your slides, please.
Yeah, we are moving.
Yeah,
Slides
now
are
it's moving. Sir, now moving.
Okay. Slide shows.
Now it is moving.
Yes, sir.
Okay. Now then this is another interesting result that endophytes has been extracted from the PGNP plants and used as a resistant material for this chickpea color rod, that is fusarium, and also it shows a drought resistance. So this is an interesting result, and it is under field validation at present. Now, this is another interesting result. You could see that in the whole world, that whitefly genetic groups are present at 44. In India, only 11 are existing, but our team of scientists, they have identified another 3 new genetic groups of whitefly, and particularly in Chhattisgarh area, where the central India. So this is a good achievement, and I think now the all characteristics and transcription efficiency is under testing. Now, Chiramon is a plant-based product, and I think if we spray to the this your trichogamma, it acts it shows a good control of the trichogamma species. Now bacteriophage, I think we understand the phase is against the bacteria. So the bacteriophage we have identified, and we are using against the rice bacterial leaf blight pathogen, and it is still I think it is under field condition. Now super donor, I think a group of scientists are looking for development of the multiple resistance using the super donor. They have identified, they have already come up with the black cross of a bacterial leaf fly, but they are looking for the back cross, and also work is in progress for brown leaf plant hopper and blast. So if we ultimately if we achieve this will be a revolutionary into the multiple stress resistant the rice crop. Yes, we understand that this direct seed technology, which is a serious problem, and ultimate aim of this direct seeded rice is efficiency. Maybe input is efficiency, but the herbicide is a problem. The weeds is a problem, so it is herbicide dependent technology. So can we go for the alternate approach and low cost or zero cost approach, that is identification of the plant germplasm which can suppress the weeds, so that weeds loss can be minimized, the crop losses by the weeds can be minimized, and then we can also minimize the load of the herbicide to the environment. Now this is another reason. You see the very interesting exploring the host microbial crosstalks in the agro ecosystem. We understand that there is in human plant relationship. The many diseases or infection is coming from either from the soil or plants. So here that how this infection is coming from soil and plant and affecting these pathogens are affecting the animals and ultimately affecting through the milk or meats is affecting the human. So this one health through one health concept, this project is going on, and I think we can identify the microbes and pathogens and which ultimately affecting the human. So we understand this is a government scheme. The government scheme is running on this One Health concept. So we are and One Health program with the climate change. Now frontier. So once this is we know that so far visual symptoms is used for early detection and diagnosis of disease and plants, the disease and pest of any plants. But here now the frontier science we should adopt. We know the various frontier sciences, that is system biology, plant defense with the emphasis on role of endosymbionts, and also genomics knowledge tools for molecular diagnostics. Now if we use this, definitely we can bring revolution in the molecular biology and bioinformatics. So this is our expectation, and with this expectation we are working on that in our Indian condition, deciphering host defense mechanism against pathogens and understanding the regulation of defense responses. We know that genomics and gene editing is a now emerging area for enhanced multiple biotic stress resistance. Work is going on. Then stress interaction. We always work in isolation, that is biotic stress and abiotic stress separately. Can we have an understanding how these both the stresses interacted each other, so that we can come a common management aspect and combine if we understand the combined stresses very well. Yes, we know that we so far the crop losses by the farmers field be due to the climatic hazards, or by any means we go for a manual estimation, or we are dependent on the state official. They give a report, manual report. Now this is now we can use the modern tools, remote sensing, or maybe artificial intelligence for yield loss, and I think work has started in Indian condition, and how we can also go for the biotic stress, the crop losses due to biotic stress, that we can estimate. Now biotic stress management in the emerging area, we know the three in our condition, in Indian condition or Asian condition, there are three emerging production system. One is a conservation oil proof system, conservation system. agriculture, and then organic farming, and then protected cultivation. My question to audience and others: do you have a sufficient data generation on the biotic stress management or biotic stress management on these three aspects? I think our answer is no, because very limited work has been done. But this is the priority area to develop or generate the data on biotic stress dynamics on three aspects. Now focus on One Health. I have already covered that what is will be the how this soil, plant, animal, and human continuum will work in through One Health approach under climate change. That for forewarning and estimates of the crop losses. As I mentioned, that so far we are using this visual symptom traditional method. Now developing a near real time surveillance technology for biotic stress management using the spatial and machine learning, and that is artificial intelligence is one of the major component. Definitely this will help in this forewarning of the transboundary biotic stress we are facing, and also we can develop the desired level of protective advisory if we understand the forewarning level. Now this is another situation that in our condition, Indian conditions so far, we do not have any national strategic crop health monitoring networking system. But once we have that, we can have a strategic crop health monitoring regularly. We can have a microbial resource center, and also we can go for analysis of the pest risk analysis. So this is also need of the hour at our in our Indian condition for developing the networking system. Now, if we develop that, we can have a planned diagnostic networking system through this networking system for early detection, accurate diagnostic, and early communication. As I mentioned, that traditionally we go for the visual inspection, but now there is a modern technology. We are using ELISA and PCR. We all understand that this is a technology. We go for early detection in the laboratory condition. Yes, we have. This is we know these are the 5 molecular techniques we are using for pathogen plant pathogen detection, where the polymerase chain reaction, that is PCR, is very effective, but we are effective as compared to others. Yes, we have also diagnostic techniques under field condition. Yes, field condition where this 2 techniques, that is a loop mediated isothermal amplification, that is LAMP, and also recombinase polymerase amplification, RPA, are the most common one, and are also working very effectively in our Indian condition in field diagnostic technique, as a field diagnostic technique. Yes, as a matter of fact, our institute has developed a LAMP for rapid detection of Mesta-Yellow-Ven mosaic virus very recently for the jute crop. So this is one of the recent development. So LAMP technique is a very effective technique for field diagnostic as a field diagnostic key. And also this is the once we achieve this, then another project we are also we just working. We have initiated the work for developing a diagnostic kit for the Phytophthora disease in the citrus. We understand that citrus Phytophthora disease is a serious disease. Can we develop a kit for this for a diagnostic kit? Now coming to the we are talking on that automation technology. Yes, this will bring the or transform the agriculture into digital agriculture, definitely. With the development of computation system and graphical processing unit, definitely now this is the need of the hour, that is the artificial intelligence, particularly deep learning system. Artificial intelligence with deep learning definitely will help to identify plant diseases. But in our Indian condition, this artificial intelligence is still an infant stage. We need a collaboration with the many stakeholders. We need the involvement of private sector, and also government's PP in a PPP mode, because it needs a holistic approach. It is not a single division work. It needs a holistic approach and participation of many stakeholders. Yes, I'm not going to read this deep learning. I think the many engineers are there, and also they can better understand. But it is develop a image for a detection of the plant disease or pest. And then, but one thing I can say, deep learning techniques are mostly the artificial neural network based, but a single artificial neural network will not work for disease identification. A bunch of ANNs stacking on one or others to make multi is required to make a multiple layer as an input layer, output layer. These multiple layers is also called hidden layers, and ultimately this makes a deep neural layer, and this is we understand as a deep learning. Now, these are the some of the deep learning is used for the plant diseases image generation of different leaves of various crops. This already work is going on. Now, is another technique detection technology. Yes, this is a sensor based, and it needs a violet organic compound. Now, when this violet compound, when it will put on the across the sensor, it this molecules change induced to change the physiochemical changes to the sensing material, and ultimately using this we can identify, yes, this disease is caused by which whether it is a fungus disease or bacterial disease or which disease it is. So it is very effective sensor based electronic nose we can use, and it is being used also. Now, biosensor based detection, this is how the CNOS works. As I mentioned that it is a antibody based sensor. When you talk of the CNOS, these antibody sensor are also this can detect the plant pathogens, and it is being used in many crops listed here. Now, this is some of the result I am showing here. I am not. This is enzymatic sensor based this sensor. Now, bacteriophage-based sensor is also possible. Bacteriophage, as I mentioned, that phase is against the bacteria. So, how this bacteriophage sensor? But this application is not that wide so far. But this can be used against the disease-based diagnostic. Now, this is the comparison that how the different sensor are effective, which one is good. Definitely nano material sensor based is really good real time. Advantage is that real time, easy to fabricate and stable, and also enzymatic biostatistics is also high specialty, but it has some disadvantage and lacuna. DNA based, molecular based, as I mentioned, biostatistics is a high cost. So it is up to us which one is we have to select based on the resources and also fund and also the farmers need. So this is something I want to share with you all. As I mentioned, I am not an engineer or biotechnologist, but as a RMP man and also natural resource man, I want to share with you. This is something I have shared with you. Thank you very much.
Thank you, Professor Ghosh, for your very good general view of biotic stress scenario in our country and the problems we may be faced with. I just have one query. You see that leaf curl virus comes to India through Pakistan from South Africa.
Yeah.
And like that, UG99 may come from Iran or Bangladesh to India. And we don't have much resistant sources to them. In first case, what we are doing to kill the white fly, and in second case, what defense sports we have for not spreading UG99 in India.
Yes, as I mentioned, very few work has been done in different lab of the our country, because as I mentioned, there is no networking system. There is a strong need of the networking system, and our institute being a nodal institute or national institute in our country, and also the deemed to be university status. We should work on that, and I think I have placed this proposal to Indian Council of Agriculture Research to identify and allow us to develop a networking system for this one. Until unless we go for that, it is a very in piecemeal work is going on, and there is no coordination. So this is something lacuna, because until unless we have a better coordination, monitoring, networking, I think only thing what is going on when there is an outbreak, when there is a transboundary pest outbreak, we formulate a committee gives submit report, and that's all. So this is the fate of our this recommendation. So until unless we have a nodal agency responsible for that, I think we cannot go much progress.
Okay. Thank you.
Thank you so much, sir, for your talk about such a wonderful topic. In continuation now, I want to introduce Professor M.V. Rajam. Professor M.V. Rajam is currently working as a UGC BSR Faculty Fellow in the Department of Genetics, University of Delhi South Campus, and has over 35 years of teaching and research experience. He has guided 37 PhD and 11 MPhil students and 26 postdoctoral fellows and handled 27 major research projects. He made several short visits to various countries, including France, Italy, Netherlands, China, Indonesia, and Malaysia, under the collaborating projects and for attending the international conferences. He is a fellow of four prestigious academies, the Indian National Science Academy, National Academy of Sciences India, Indian Academy of Agriculture Science, and Telangana State Academy of Sciences, and member of several learned scientific societies. Now I invite Professor M.V. Radam to present his views about RNA silencing for control fungal disease in crop plants. Welcome, sir.
Yeah, thank you for your kind introduction. Let me share my presentation.
Yes, sir.
Right now I'm using my mobile. I don't know how to share it. Let me see from here. Yeah, share. Okay. Yeah, how to go to my files in the mobile? You have any idea?
Just share your slides.
Yeah, I've already pressed the share,
share option.
and then the files are appearing.
Yeah. Files. Sir, you have not shared actually. We are
No?
not getting. We are not getting at
Yeah.
screen.
No, I'm not finding PPT. In the. I'm not finding my files. I've saved,
Sir,
PPT
just
in my
open
files.
that file on desktop.
No. Okay. Let me see.
Then you have one option to share at bottom of
Yeah.
StreamYard.
Yeah. Just wait. Yeah. Now
click
I
on that
found my
option.
file. My files. Yeah, I can find. Sorry. Just wait. Wait.
Okay.
I'm also opening a laptop. Yeah. Actually, I was using tab initially, and there was some problem. The net problem. Yeah.
Sir, kindly on your mic and camera.
Yeah. I think. No, I think.
Sir, his device is not connected. We are not connected to.
As we are not connected to Professor M.V. Rajam, we, we have reached to Dr. Tai Girma. Dr. Tai Girma is PhD in Computer Engineering from University of Ulm, Germany, and AASTU, Ethiopia, MSc in Computer Engineering, Addis Ababa University Technology Faculty, Ethiopia, and BSc in Computer Engineering, Defense University College, College of Engineering, Ethiopia. He is Deputy Director General, Ethiopian Artificial Intelligence Center, and prior to this, he was Assistant Professor Addis Ababa Science and Technology University. He is author of many research papers and books. His area of research is pattern recognition, deep learning, digital image processing, and big data analysis. Now I invite Dr. Tai Girma to present his view about past and current status of AI, artificial intelligence in Ethiopia, and current projects for collaboration. Sir, please welcome.
Thank you, madam. I hope everybody is active. Being a last presenter is not that much easy. And also, please be informed that I have a very unstable internet connection, so I will try to be as fast as possible. Okay. All right. I'm trying to share my screen. Oh, okay. Are you able to see my screen? I guess. I have the same problem. I'm unable to share my screen.
Sir, kindly open your PPT on your desktop or laptop, then
Yeah,
share from StreamYard.
I do have that. I do have already. I have opened it. I'm trying.
Okay.
Sir, please unmute your mic.
Okay, sorry. Are you able to see my screen now?
Yes, sir.
No, sir.
But
Actually, you have shared all your entire screen. Kindly share that window only, PPT window
Okay,
only. Now it's visible, sir. Now it's right.
okay, good.
Okay. Now you can continue, sir. Please.
Okay.
Okay. Thank you.
Thank you so much again. As Madam mentioned, I am currently a Deputy General Director of Ethiopian Artificial Intelligence Center, and I will present to you the past and the current states of AI in Ethiopia. And there are also projects which since we are a late comer to the area, there are projects that we have identified for collaboration. As you see, Ethiopian AI sector has a very few players. Can you hear me? Hello?
yes, sir.
Hello?
Yes, sir. We are hearing.
Okay. Okay, I just want to make sure that everybody is listening to me. So the Ethiopian AI sector has a very few players, and all those players are from the private sectors. One is the Ethio Cloud, which is a system of cloud-based API developed by US-based company. And Gabianet is also another software development outsourcing provider with a client across the United States, Europe, and several African nations. Again, there is also an EthioRobo and ICOG Labs, and these are all the private companies which has running smaller projects in Ethiopia. Of course, there is also another company which recently announced. It has been, since its establishment, it has been working as an incubation center for AI and ICT. And from the government institute, we found that there is a center of excellency for AI under Addis Ababa Science and Technology University, which aimed to work in creating a close collaboration between academia and the industry in the field of AI and robotics. And there are also other more smaller companies, of course, which are private, like Abogida, Faris, and other smaller privates are there who has been a player in AI industry in Ethiopia. However, all of them didn't impact the nation with AI application. This is because of a different challenges that we recently identified before the establishment of Artificial Intelligence Center. One of the reasons was all of them were working independently. And of course, when we talk of artificial intelligence, then it is all about the data set. So if you, as some of the presenter, I think I can, if I'm not mistaken, Dr. Tino was telling that data preparation is very expensive. Yes, that is what we have already identified as a challenge in Ethiopia. Of course, during a data preparation, the existence or the involvement of the domain experts are very important. And of course, the costs of those domain experts are also untouchable. The other challenges that we have seen of reason for applying AI in Ethiopia becomes so difficult is that there is no any centralized data set available, even those which are not structured or well annotated. Another reason is there was no any computational intensive resources for big data processing in Ethiopia. Of course, not least, but incentivizing the researchers to be moved into this area is also not existence and it has not been there in place. So these are also one challenges that we have seen. And of course, another reason that we have identified is there is a lack of financial support for the AI researchers. And that is why some of the private companies who has been a player in the area has been closed because of even covering their monthly salaries. And then there is no any national hub in the country for organizing and facilitating and supporting those private companies, and also motivating and encouraging the public institute to work in the area of artificial intelligence. So what we have identified that the national center is very important, which can be a national AI hub to elevate these challenges and encourage the private companies and, of course, the public universities to work in the area of artificial intelligence. So for that reason, in 2020, His Excellency, our Prime Minister, has visited the United Arab Emirates, and he has got an opportunity to see how artificial intelligence has impacted the life and the daily activities and the services of the United Arab Emirates. Right after he get back to Ethiopia, His Excellency Prime Minister Dr. Abiy has initiated the artificial intelligence center to be launched. And so with the same year, in 2020, through the councils of the ministry, the Ethiopian Artificial Intelligence Center has been established. And then September 20, in the year 2020, His Excellency Prime Minister Dr. Abiy and our General Director at the right has inaugurated the center. And you can see on the left bottom corner, you can see me while I'm just presenting our Prime Minister what we have been doing since the establishments of the center. And so the center has been established with a mission of fostering the development of nationally recognized AI ecosystem to empower and inspire a nation for peace and prosperity with the most trusted analytics. The vision we want to be in, we have a 10 years vision, and we want to be a state-of-the-art national AI research and development center with an excellency, and of course with a key role in creating innovative AI-enabled solution at the national and international level. So we have set 3 objectives. Hello? Hello? Hello?
Hello.
Yeah, I just, because of my unstable internet connection, I just want to make sure that still I am alive.
Oh, you are alive. We are listening you very carefully. Each
Okay,
word.
okay. Thank you, Professor. Thank you.
Yes.
So the center has set 3 objectives. One is to be a center of excellency for the research and development in the AI. The second objective, since we are a latecomer and no such organizations in the country, so if we are going to work on the AI, then there is a need for national AI policy. And so the second objective is we want to set a policy and infrastructures framework for the development and the deployment of artificial intelligence. And the third is we need to create an ecosystem for artificial intelligence startup. And that is actually we are doing currently. We said we were preparing in April 15 to 18 the workshop which connects the academia and the industries together. And of course, we have also today expos in connection to the workshop. Following the workshop, of course, we want to encourage individuals and the private companies to be visible to the public, and also creating an opportunity for supporting them financially in a different way. What we have achieved so far in the area of the sector, we have developed a prototype, a breast cancer detection system, which is actually a continuation of my PhD research work. I hope Professor Hakim he has been my supervisor while I was doing my PhD, so he knows this research. And then in the health sector, again, we have developed a prototype brain tumor detection system. Again, in the health sector, as earlier I said, that data is very important in the area of artificial intelligence. So we were trying to collect the data through developing a teledermatology. Of course, finally, this time now we change it into the telehealth system. So in the telehealth system, there are a lot of services that we are planning to do. In the meanwhile, in the meantime, we are also while giving the services, we will be able to get the data from the telehealth system. In the finance sector, we have developed an AI-enabled customer support service chatbot for banking industry, which has covered 3 local languages and 1 international language, English, Afan Oromo, and Amharic. These are the 2 languages. Amharic and Afan Oromo is the most widely spoken language in Ethiopia. So while doing this, there is also some attempts that we have been doing in language. We have selected 4 Ethiopian language, and there are a lot of audios, and those audios has been undergoing for transcription, so that localization, any artificial intelligence technology that we develop has to be localized into the local language. So for that reason, in the center, we are doing an amazing job in transcribing those 4 languages. Actually, these languages are Amharic, Afan Oromo, Somalinya, and what we call Tigrinya. These are the 4 major languages spoken in Ethiopia. Coming to the last, we have research projects which we have identified in the health. These are the projects that we have identified. Of course, these are very open to work with an international institute in collaboration. So the breast cancer detection and the management, of course, it is also under research and under development. Brain tumor, and all these are aggressively we are collecting. We are assigning a budget for this to collect these projects to collect the data, and a prediction of diabetes and its complication management, and a cuff sound analysis for pneumonia and the TB. We are also trying to work it at the smartphone level. Of course, this is going to work for a COVID detection also, which of course we haven't started working on that, and of course this, if we are able to achieve the pneumonia and TB diagnosis through the smartphone, and that the same way we can do for a COVID-19 detection. And of course, another project that we have identified with the hospitals is ECG interpreter. We have a very limited cardiologist here in Ethiopia. For example, I can mention a neurologist. We have only 1 neurologist in the country for the 100 plus million population in Ethiopia. And the other is we have most common skin diseases in Ethiopia, which majorly affect the children. So on that, we are also aggressively collecting the data to develop a deep learning model which can detect and enable us to manage the diseases. And of course, I have already mentioned that we are also working on that telehealth system. With the finance sector, we have developed a customer support service chat for banking industry, but still the credit scoring in Ethiopia is subjective. We want that to be objective, and we are also working with the major banks, Ethiopian Commercial Bank, and 1 private bank, Cooperative Bank of Oromia, on the credit scoring. And with agriculture sector, we are planning to work on this, and in some of the area we are collecting the data. Inset is sometimes we call it a false banana. It has no any fruit, but 20 million of the Ethiopian population uses as their main food. So working on this area, but there is also a disease which we call it insect bacterial wilt. So dealing with this disease is going to be worse enough. So we are also trying to work on that. We have some preliminary studies, and we have published also 1 paper with one of my MS students. And the rose disease detection and the management, rose, especially during this COVID-19 cases, 20 to 30% of export was rose, while other companies, other neighboring countries didn't do that. We were able to secure like 20 to 30% of hard currency generation in this area. Cereal crop disease detection is also one of the reason, because maybe Dr. Yemisrach has told you in March 8th presentation, around 40 to 47% of the GDP of the country will be supported by agriculture. And so working on the disease management, detection and the management on the agricultural area is going to be also very important for us. At the moment, we are doing coffee disease detection and the management, but all these, for example, like a cash crops, sesame, and of course, pulse crops disease detection is also very important. And livestock diseases are also one of the major challenges that we have in Ethiopia. So we are working on this, and so we invite the institute who are very willing to work with us. And finally, in the transport system, we have identified a challenge in Ethiopian transport system. So we are planning to work on also intelligent transport system in Ethiopia. Thank you so much for taking the time with us. Hello.
Hello. Thank you, Dr. Germa.
Okay.
Hello.
Hello. I'm listening.
I would like to suggest you to first work on simple systems like anthracnose in pulses. Okay, they are bacterial diseases. Their symptoms comes on surface, and then thereafter you go deeper and deeper. Okay, with a simpler system like in NSET also. But the coffee rust will be very interesting. The phyllodes in sesame, the viral disease in sesame will be quite interesting because your sesame quality in Ethiopia is the best quality, and that should remain so that you can get better uh, returns from that. Okay, so you can start with the plant diseases detection on simply inherited diseases and very conspicuous diseases like leaf blight, white rustling brassica, and bacterial blight in case of pulses. So these are some of the diseases you should target. They are important for you because of the financial reasons. These crops are being exported from your country. So that is what is my suggestion. And you should submit a proposal to either EU, we will discuss after this webinar is over, or to McKnight Foundation in USA. They are Africa centric. And the biggest funding from McKnight Foundation. Okay.
Thank you, Professor. Your point is very important, and then we will work on it together. Apart the disease, we have also a challenge in sesame that a maturity level is not easily identified, and some of the yield loss is there with the sesame. So we can also include.
It can be standardized. It's not big problem. It can be standardized by AI ChatGPT. That is what and Surya has suggested. That we can identify the chlorophyll degradation and standardize a stage that when chlorophyll degradation is this much, harvest the crop without waiting.
Okay,
that is
thank
the
you.
Easiest, that is the easiest to do. Okay?
All right. All noted. Okay, thank you so much.
Okay.
Thank you so much, Dr. Girma, to make all of us aware about all these valuable things. Now I invite Professor M.V. Rajam again to present his views about RNA silencing for control fungal disease in crop plants. Welcome, sir. Yes. हाँ.
अच्छा इधर से कर दिया क्या?
हाँ, सर. Sir, unmute yourself. Your mic is muted. 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 Sir, unmute yourself Sir, mic is kindly unmute first.
Can you hear me now?
Yes,
Yes.
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 Yeah, so you can go for slideshow, the presentation.
Yeah.
Yeah.
Ma'am, visible?
For full screen,
Yes, sir.
but
First
it is
Yes.
not showing. Huh? It is not showing full screen.
It's a
Not
full
sharing full
screen.
screen.
No, you can see full screen.
Slideshow.
No, I don't think.
Yes,
I
yes,
don't
we
think.
can see.
Sir,
Oh,
Yes.
then it's okay. No problem. Yeah.
yeah.
Okay.
Sir, tell me when
Yeah. Yeah.
I
Sure.
need to go to that, please tell me.
Pardon?
When he needs to move the slide, please tell him. Next.
Yeah. Sure. I'll do that. Yeah.
Okay.
Yeah. So at the outset, I would like to thank Professor Bell and Anuj Rana and the other colleagues there for the invitation, and also sorry for the inconvenience caused in opening or sharing PPT presentation. So we have lost few minutes because of that. Anyway, so today I shall be talking about RNA interference, a relatively more recent technology, which is being used for the control of fungal pathogens as well as control of viruses, bacteria, insect pests, and nematodes. Apart from its potential application in agriculture for improvement of various traits, including nutritional improvement, so oil quality improvement, etc. Next, please. Next. Yeah. Next slide. Hello. Next slide, please.
Sir, I'm giving next.
Hello.
Yes, sir. Yes, sir. We are at second slide.
But I can't see second slide here. As I'm still seeing the first one. Yeah. Now it's okay. Yeah, it's fine.
Now, sir. Now it's
Yeah. Now it is fine.
yeah.
Yeah. The RNA interference phenomena was discovered by these two guys, Dr. Andrew Fire from Stanford University and Dr. Craig Mello from University of Massachusetts. And their pioneering work was published in Nature in 1998. And this is one of the most important breakthrough discovery after discovery of DNA structure by by Watson Crick. And RNA interference is basically the sequence-specific gene silencing, which is mediated by presence of double-stranded RNA. So how they have demonstrated that double-stranded RNA has made gene silencing. I'll give you only one example, although they have worked on more than 20 genes for their silencing using double-stranded RNAs. So look at the bottom figure. They could suppress the expression of a gene called UNC22 in C. elegans. The expression is based on immunoprecipitation technique. And look at A. A is a normal control. B is a user expression. And C, they have imbibed C. elegans worms in antisense RNA, but they could only see a very partial suppression of the target gene UNC22. The last one, D, when they incubated C. elegans worms in the presence of double-stranded RNA against UNC22 gene, and they could see almost 100% silencing of UNC22 or target gene. So based on this observation, they said double-stranded RNA made gene silencing, not a single-stranded sense RNA or antisense RNA. Anyway, it is also involved in gene silencing, but not to that extent. So that means the double-stranded RNA mediated gene silencing is much more effective, more than 100-fold effective in silencing the target gene as compared to antisense RNA. And they have also observed a couple of more important things. They said the gene silencing can pass through from one generation to next generation. Not only that, the silencing signal can also move from one cell to other cell, right? Which is facilitated by some proteins. And of course, for this important discovery, they have got Nobel Prize in 2006 within the span of 8 years. And now we know that RNAi pathway is highly conserved across most of the eukaryotic systems. And in fact, RNAi, including siRNAs, small interfering RNAs and microRNAs, they are very much involved in the regulation of gene expression. Until recently, people thought that the gene regulation is only mediated by, through certain phenomena, transcriptional, post-transcriptional mechanisms. But now it appears that small RNAs like microRNAs and siRNAs, they are very much involved in regulation of gene expression. So they are very important molecule now. And RNAi is also involved innate immunity, particularly against viruses. And people believe that this is one of the ancient innate immunity mechanism against viruses in animals, humans, and plants. Next, please. Next slide. Yeah. Yeah. So let me take a minute to explain the general mechanism of RNAi-mediated gene silencing. So this pathway has basically two steps, dicing and splicing. So the triggering mechanism for RNAi is double-strand RNA. And double-strand RNA can be formed in the cell by a variety of ways, like viral infections, the introduced foreign genes or transgenes, or read-through transcription of multiple genes, or presence of aberrant messenger RNAs having secondary structures that can also lead to double-strand RNA in the presence of RNA-dependent RNA polymerase. Or you can also prepare RNAi hairpin constructs by cloning the target sequence in sense and antisense orientation downstream to a promoter. And after transcription, this can also lead to formation of double-strand RNA. I'll be explaining after a while how we prepare the hairpin RNA construct or silencing constructs. So that means once eukaryotic cell experiences the presence of RNA, double-strand RNA, then it triggers the gene silencing mechanism. So initially the double-strand RNA is cleaved by RNase 3 enzyme, that is dicer, into very specifically 21 nucleotide size. And these small RNAs of 21 nucleotide, they are called small interfering RNAs or siRNAs. And siRNAs, invariably, they have two important features. They have two nucleotide overhang at 3' and phosphate group at 5'. And now these small interfering RNAs, they are recruited or they are recruited a multi-protein complex called RISC, RNA-induced silencing complex, where the argonate protein is one of the important in this complex. Argonate protein also belongs to RNase 3 family of enzymes. And then the RISC gets activated, and argonate protein, it selectively degrades the sense strand or passenger strand of siRNAs and retain only the antisense, which is also called guide strand of siRNA within the RISC complex. Now the activated RISC complex along with the antisense RNA can identify and cleave or degrade the cognate messenger RNA or target messenger RNA. And as a result, there won't be any protein. So that means the target gene silencing is happening after transcription. So therefore, this pathway is also called post-transcriptional gene silencing, which is nothing but RNAi. So as I said, the pathway has two steps. Dicing means the formation of siRNAs by the cleavage of double-strand RNA by dicer. The slicing means degradation of target messenger RNA by argonate protein with the help of antisense siRNA. And interestingly, after the formation of small RNAs, these can also pass from systemically, this can also move from one cell to other cell with the help of seed proteins, membrane-bound seed proteins. And another feature is the siRNA molecules can also get amplified to form a secondary messenger RNAs. And this can happen, after the cleavage of target messenger RNA, the bigger fragment of messenger RNA is acted as a template, and the single-stranded siRNAs act as a primers for the polymerization of the messenger RNA fragment in the presence of RNA-dependent RNA polymerase. That is how we see formation of secondary siRNAs. Next, please. Next slide. Next slide, please.
The next slide.
Yeah, okay. So let me explain how we prepare silencing constructs or hairpin RNAi constructs, because these constructs can be used to target or to silence any gene in different eukaryotic systems. So initially, you have to find an important target gene for the control of fungal pathogen, let's say. And then after having identified a target gene in the pathogen, you can retrieve the sequence from NCBI, and then the sequence can be used for blast search just to select a region of the target gene which has minimum homology. So that means that a fragment, target gene fragment or partial sequence having low homology can be used for cloning in sense orientation, as shown in red color with arrow, in sense orientation as well as antisense orientation, with a small interfering DNA in between them. So generally we use a small functional intron as a spacer DNA between sense and antisense gene, and these genes are under the control of an appropriate promoter, like constitutive promoter or inducible or tissue specific promoter, depending on our objective. And then of course you have a poly A at 3 prime. So if you introduce this construct into plant systems, following transcription, you get a long double-strand RNA with a hairpin. The double-strand RNA is formed because of the sense gene. It forms sense messenger RNA. Antisense gene forms antisense messenger RNA. Because of their complementary nature, they form double-strand RNA. But the intron, having no complementary sequence, it forms a small hairpin. So that is how we get double-strand RNA with a hairpin. And once the double-strand RNA with a hairpin is formed, it triggers the RNA mechanism, which is later involved in silencing of the target messenger RNA. So the silencing constructs can also be prepared by different ways, two different ways, but most people prefer this method for preparing hairpin RNA construct, which are much more effective than other methods. Next, please. Next. Yeah. So in my lab, of course, my lab is one of the first labs in India to initiate two major programs on RNAi for the control of fungal infections in tomato, particularly Fusarium oxysporum, that is Fusarium wilt, which is a very important disease in tomato, and also the control of fruit worm, that is Helicobacter armigera in tomato. So these programs were initiated in 2005, and then later, of course, we have done very significant work in this area and published quite a few papers. And for control of fungal plant diseases, so we are using two different approaches based on RNA interference. The first one is small interfering RNAs or sRNAs, and more recently, we're also using artificial microRNAs, that AMI-RNAs. Next, please. Next. Yeah. So, oh no, the previous one, the previous slide. Yeah. So RNAi has already proven to be a very novel and potential alternative for crop protection against various pathogens and various pests. And in fact, RNAi is also being used for crop improvement, for improvement of many agronomical important characters. So I'm not getting into details because of paucity of time.
Yes.
Yeah.
We will organize your lecture once again.
Yeah, sure. So what is important, for the control of fungal pathogens, what is important is the identification of a novel target gene in the pathogen. And that identified gene's product, it should be absolutely important for fungal growth and differentiation or development. So therefore, if you knock down the expression of that identified target gene in fungus, it will lead to lethality, because after silencing of the target gene, fungus cannot grow any further. So that is how we can control the fungal infections of plants. So when we initiated this work in 2005, we had very few such important targets for RNAi. But now, since we have complete genome sequencing sequences in different organisms, including plants, fungal pathogens, etc., so identification of novel target gene for RNA is not at all a problem. So you have a depth of targets for the control of fungal pathogens by using RNA approach. Next, please. Yeah. So selection of a target gene from fungal pathogen. Where several years ago, I've done a very significant work at Yale University. So there, we have targeted polyamine biosynthetic enzyme, ornithine decarboxylase, which is served as a novel target for plant chemotherapy against fungal pathogens. In other words, we had used a specific inhibitor of ornithine decarboxylase enzyme, that is difluoromethyl ornithine, and by spraying a very little concentration of DFMO onto pinto beans, we could control the pinto bean rust, that is Uromyces faciali, very, very effectively. You can see yourself in the photograph. On your left, DFMO treated and protected pinto bean plants, and on the right side, of course, the badly affected control plants by the Uromyces. So this work was published in PNAS in 1985. Since then, of course, a lot of groups have repeated this work for the control of various other fungal pathogens. So based on this work, I thought that ornithine decarboxylase might be a novel target for the control of fungal pathogen using RNAi approach, right? And here we have used the inhibitor of a ODC enzyme, but we wanted to try the RNA approach targeting ornithine decarboxylase or ODC gene of target fungal pathogen, that is Fusarium oxysporum. Next, please. Next. Yeah. I think this slide, I've depicted the concept of RNA for the control of fungal pathogen in a single slide. As I discussed briefly before, the first and foremost important is the identification of a target gene. Here, of course, we have identified ODC, and then the preparation of hairpin RNA construct by cloning partial gene of a fungal ODC gene in sense and antisense gene under the control of a constitutive promoter. And then we use this construct, hairpin RNA construct, for tomato transmission by agrobacterium. And now the RNAi tomato transgenic lines express double-stranded RNA and subsequently form siRNA molecule. So that is what is happening in the plant, RNA lines. When such RNAi transgenic plants are infected with the target pathogen, that is Fusarium oxysporum, we expected that siRNA molecules which are being synthesized in tomato might enter into the invalid fungal cells. So after entry of siRNA molecules from transgenic tomato plant into fungal pathogen, now the siRNA molecules are recruited or uses fungal RNAi missionary. That means the siRNA molecules are recruited in fungal RNA RISC complex and then target or silence its own gene, ODC gene. That's very interesting. That means you are silencing a fungal gene via or through RNAi mediated host mechanism. So that is how we see silencing of the target ODC gene in fungus, and as a result, we might see fungal resistance. So with this hypothesis, actually, we have initiated this work in 2005, and we have got very exciting results. Next, please. Next one. Yeah. So let me discuss very briefly what we have done. So as you know, Fusarium oxysporum causes Fusarium wilt, and these are the typical symptoms. Next one. This work was done by Neeru Singh. Next, please. So initially, we have retrieved ODC gene from Fusarium oxysporum from NCBI, and then the sequence was subjected for blast search. Then we selected about 560 base pair region of the target gene, which has very low homology. And then again, this 560 base pair sequence was divided into 21 base pair fragment, because the target sequence, fungal ODC gene sequence, will form multiple siRNA molecules of 21 base pair size. So we wanted to rule out any off-target effects of every siRNA molecule of 21 base pair size. So that is the reason the sequence was divided into 21 base pair fragment, and each of these 21 base pair fragment were again subjected for blast search, and just to know or rule out any off-target effect, that is silencing of unentered genes. And then we have also checked the prediction of putative siRNA molecules from the selected sequence. And finally, of course, we have used M-Fold software to predict the stable structure of hairpin. So having done all this exercise using in silico analysis, we found that the selected sequence of 560 base pair has minimum homology, and it can form potential siRNA molecules, and it can also form stable double-stranded structure, and as shown on the right-hand side. Next, please. And then, yeah, so then we have cloned 560 base pair sequence of ODC from Physarum oxysporum by by using cDNA. Then this sequence, 560 base pair sequence, was used to prepare hairpin RNA construct. I've already discussed, how we prepare hairpin RNA construct. And then this construct was used to develop more than 60 tomato transplants by agrobacterial transmission, and then the potato tomato transplants were screened by PCR to demonstrate presence of transgene, and more than 70 tomato transplants were positive for presence of transgene. Next, please. We have also done southern analysis and all to know the copy number of the transgene, but I'm not showing all that. What is important is detection of intended siRNA molecules of 21 base pair size. So for this, what we have done, we have enriched small RNAs from isolated RNA, and the small RNAs were used for northern hybridization using ODC as a probe. And as you can see here, most of the tomato transgene clients have shown a very good titer or expression of siRNAs. And then we picked up some of the promising lines like 80, right? Different 10 number, 80, and so on. And then these lines, selected lines having or showing the very good expression of intended siRNA molecules were challenged with the spores of Physarum oxysporum. And actually, we have used two different methods for infection assays. So the first case, what we've done, we have dipped the root system of control and also root system of different RNA lines in spore suspension of Physarum oxysporum for about 20 or 25 minutes. After incubation, we have established seedlings in pot condition. Or alternatively, one can also go for, this is called a root dip method. And then, as you can see, in the case of control, as you can see, there are wilty symptoms after 10 days or so. But the different RNA lines, they have shown different degree of resistance against Physarum wilt. Next, please. Next, please. Yeah. We have also calculated disease index based on 5 scale. Control, of course, if your value is up to 1, that means highly susceptible to Physarum wilt. And different RNA lines, they have shown less than 1, but some are very close to 0.1 or 0.2. So those are lines like 10, 81, right, 87, so on. They are highly resistant to Physarum wilt as compared to line 32. So that means we have seen different degree of resistance depending on the expression of siRNA molecule. And in fact, the disease symptoms are also delayed by 5 to 6 days in some of these lines. Next, please. Yeah. So Physarum oxysporum being a root pathogen, because it enters the root system and establishes itself in the vascular system of the root and blocks the vascular system. As a result, we see wilt symptoms. So we wanted to verify or check the mycelial colonization on the surface of root of uninfected control or infected control and transgenic lines. And of course, uninfected control, we can't see any mycelial colonization, but infected control, as expected, there was very profuse mycelial colonization in case of infected control. But the case of RNA lines, tomato lines, we hardly see any mycelial colonization. So that indicates that even entry of pathogen is restricted right at the root surface because of entry of siRNA molecule and silencing of target ODC gene in the fungal cells. Next, please. Next one. Yeah. Yeah. So of course, that based on disease resistant assays, we predict that siRNA molecules might have entered into fungal cells. But we wanted to demonstrate the uptake of siRNA molecules by fungal cells from tomato transgenic plants. So for this, I've designed an interesting experiment. So what we have done, we have transformed Physarum oxysporum with green fluorescent protein, GFP user construct for its overexpression. And then you can see, we could see a very good expression of GFP based on confocal microscopy. And next, please. Simultaneously, we have also developed tomato transgenics with with the RNAi GAP, right? That means these tomato transgenic express double-strand RNA against a GAP gene. In case of fungus, we have used only GAP, but in tomato, we have expressed double-strand RNA against GAP. So, and then we have also checked to detection of siRNA molecules by Northern. Next, please. Then what we have done, we have challenged. Yeah. So we have challenged the control and different RNA lines with GAP expressing spores of Physarum oxyspore. And then after about 15 days, we have checked GAP expression in the infected root system. In case of infected control, GAP expression was very high, as expected. But in different RNA lines, the GAP expression was significantly reduced. So this suggests that siRNA molecules are really entering into fungal cells after infection from tomato transgenic lines. And this work was published in Plant Molecular Biology report. No, it's already published, not in press. Next, please. Then later on, subsequently, we have identified more than half a dozen important targets of the fungal pathogen, like CS, for example, charismate synthase. These are novel targets as compared to ODC, because ODC gene is also present in the plant, host plant, in animals and humans. So that time, of course, as a proof of concept, I've selected ODC as a target, but ideally, the target gene, fungus target gene, should be only present in the fungal pathogen and absent in the host plant and human, animals, and all kind of beneficial organisms, just to avoid off-target effects. Next, please. And more recently, I'll take only a few minutes, 3 minutes or so. Very recently, actually after my retirement or superannuation, I've got to UGC BSR faculty, and this is the project which I've given for BSR faculty fellowship. So we are already used artificial microRNA based approach for the control of tomato leaf curl virus in tomato. We have come up with very highly resistant tomato lines. And then there's hardly any work, to the best of my knowledge, there's only one publication where somebody has used artificial microRNA for the control of some other fungus, right? And here we wanted to explore the artificial microRNA based approach for development of tomato resistance against alternative reptile, which is also a very important disease in tomato. What are artificial microRNAs? MicroRNAs, we all know, these are also non-coding RNAs of 21 to 24 base pair size. So artificial microRNAs are involved in gene regulation for the gene silencing. So the artificial microRNAs, they are nothing but microRNAs where we replace the native microRNA duplex from a natural microRNA precursor, right? You can see in the diagram on the extreme left figure, the arrow that I've shown is the actual microRNA duplex region, which is actually going to form mature microRNA after processing. So this region is replaced with the sequence of urine trace. If I want to target, let's say, chitin synthase gene, which is of course supposed to be a novel target gene in the fungus, I replace this microRNA duplex region with a 21 base pair sequence of chitin synthase gene of Physarum oxyspore, and then now it has become artificial microRNA, right? And then interestingly, the artificial microRNA can also undergo a similar process to form mature microRNA. Then mature microRNA is then involved in gene silencing, target gene silencing. Next, please. So here in this project, I have identified a couple of targets: chitin synthase and MAP kinase. Like I'm just showing one gene now. So we have retrieved chitin synthase gene of Physarum oxysporum from NCBI, and then sequence was subjected for a software program called WMD3. These programs will generate potential artificial microRNAs of 21 base pair size. And then we blasted 21 base pair fragments of the generated potential artificial microRNAs just to rule out any off-target effects. And then we have selected one potential artificial microRNA which was generated through the software, and then replaced the native microRNA duplex region with a sequence of our interest, and then stability of this sequence, artificial microRNA, was also checked by M-fold, right? So this exercise is very important to rule out any off-target effects. And then, of course, the work on preparation of artificial, we have already custom synthesized artificial microRNA, the harboring chitin synthase gene of Fusarium. So right now, the work is in progress on the preparation of artificial microRNA to target chitin synthase of the fungus, and then later on we'll go for development of tomato artificial microRNA lines, and they are testing for resistance. Next, please. I'm almost through. Yeah. In conclusion, we have demonstrated that polyamine biosynthesis is a potential target for plant chemotherapy against fungal pathogens. RNAi, including artificial microRNA, based on my experience, would offer potential applications in agriculture to develop crops for protection against pathogens, or including fungal pathogens. And this has really opened up a new era for crop protection and also crop improvement. And I strongly believe that RNAi would complement with the existing crop protection and improvement strategies. And in fact, RNAi as well as artificial microRNA approaches have several advantages for crop protection against pathogens and pests, because here in RNA lines or artificial microRNA transient lines, there won't be any protein, no transient protein or no foreign protein. So obviously, that means the host protein machinery is not at all utilized, and in the presence of transient protein in RNA lines, it is very unlikely that the target fungal pathogen would gain resistance.
Very
right?
good.
The usual transgenic approaches, you have the problem. You have a transient protein, so the target pathogen there's a possibility that target fungal pathogen can gain resistance. And I would like to acknowledge my research team. You can also see Dr. Anuj Rana on the left-hand side, second from left-hand side. And most of them have already completed their PhD or postdocs. And I would like to thank the funding agencies, particularly Department of Biotechnology, who have generously supported my RNA program, several RNA programs, and my collaborator, Dr. Sunil Mukherjee, who had helped in the fungal work. Thank you for your patience.
Thank you.
And I'm happy to take up any questions or queries.
We are running short of time. We will organize a lecture once again, exclusively on this topic, and international webinar. So
Yeah,
then
sure.
we will have enough time to discuss. Yeah.
Yeah, sure, definitely.
Okay.
Yeah.
Thank you.
Thank you so much, sir, for such a good information. Now I request all the speakers to join for panel discussion.
Sure. Okay. Thank you. Thank you, Dr. Bell.
Yeah. Welcome.
Yeah.
Welcome. इधर है मदन की बीसी से बोला ना हाँ मेरे को मैं बोल हाँ VC sir, we can ask him also to come on the board. We will ask him even to come on the board for a while. And meanwhile, our purpose is to initiate some discussion regarding the possibility of multinational projects. So in this direction, I will request Madan Thengevalu to please initiate the initial discussions.
Thank you. Well, sir, thank you so much, respected Sunil Singhji and Tai Girma, Rajanji. Thank you so much for all your wonderful presentations. Greetings from Cambridge. It's a delight to be here at this event, and what has come through in the last 2 days is a wish for bringing out the power of new technologies. And I think in Tai Girma's presentation, this came through very nicely. The power of new technologies to be used in new environments, the challenges and how to use it. Number two, the point that came through very importantly in Tino's presentation, the concluding slide, which is there is a need for having more minds who can analyze these problems critically and this critical analysis is very important. And there is a need for crossing disciplines, and as Samir Singh ji mentioned, the AICT they have announced that the requirements for joining engineering programs is going to be slightly more liberalized, and this is an indication that we have to come towards um, many of these areas with an open mind. And uh, we also touched on one area of One Health, and this whole area of how to look at health slightly more holistically.
Thank you, Madan. What role you see for yourself in this proposed multinational cooperation?
What I'm presenting here, the vision that a small organization like ourselves, the OM Lab, which is based in India, with contacts across Europe, how we can engage with the universities here in Haryana, in Badurgarh, and our host institution and my alma mater, Haryana Agriculture University, and with Akim's connections in Germany, in Tübingen, and with TAIA's organization in Ethiopia, how we can very easily come together to outline concrete research areas.
Yeah, thank you, Madan. Thank you, Madan. Let us hear now. I think even though we're running short of time, and we will continue this discussion later on. But initial, as always, I will come up with another webinar only for discussion of the project. Okay. Now, Achim, even Thal.
Yes, okay. Thank you. First of all, Rishi, thanks very much for organizing this 2 days of very dense and qualified information. And thanks to all the speakers that we had. And I'm grateful for all the contributions. And with respect to the project of Dr. Taye, I think we have a lot of input and information. So I agree that we have to just put together the contacts and form a project that covers all the aspects. And many of us have mentioned different aspects as it is clearly a multidiscipline project. And so I think in the beginning we should collect some names. We should form a smaller cell of experts from India, from the UK, from Australia, whoever wants to participate and contribute. And from that, we should agree our project content. And next thing will be, of course, to look for the funding of such a project as Dr. Taye has been talking about. And especially also, I would love to involve as much experts from Ethiopia also, and maybe next to it, Dr. Taye can comment also, Rishi.
Yeah, Dr. Taye Girma, please. Thank you, Achim. Dr. Taye Girma, please.
And thank you, Professor Rish, for organizing such an amazing and a fruitful workshop. Yes, I do agree with what Professor Hakim has said. If we want to be fruitful and effective, collaborating is somehow which is not going to be an alternative. We need to collaborate. There are experts in India, in Germany, and of course in Ethiopia, even though we are very limited in the area of artificial intelligence, but we have a very rich and senior researchers in agriculture, and of course another domain. So collaborating will make us fruitful and effective, and we are very ready for that.
Very good. Very good. So now,
Professor Rish?
We request our Vice Chancellor, Professor Samir Singh, to tell our own side decision to enter into this multinational project. Professor Sumer Singh, please.
So first, let me thank everyone, Professor Madan, Professor Rajan, Professor Achin, Dr. Thai, and many other speakers who are not right now in the panel discussion to make this 2 days webinar, and that is to international level, a great success. Since I come from engineering background and I love to listen, love to learn, love to see such interactions where I think the future needs are taken care of, and I feel myself as the happiest academician that under the leadership of Professor Bahal, he is not leaving any stone unturned to connect all the, I think, subject experts and really interdisciplinary areas and connecting dots, especially to the agriculture science. So I'm really happy, and yes, I think let me express my gratitude and heartfelt thanks on behalf of the whole university team that you have spared time, all the renowned speakers, scientists, and looking forward for such sessions more in number and very frequently. But now let me come to the, I think, the last part of this discussion, and that is something in form of a project or a mission mode activity. So let me share with you that as university, we are always open for such research cooperations, and we welcome international collaborations because foundation of this university is let down on vocationalized courses. So maybe we don't believe on imparting theory, doing basic research, but at the same time, our core area is that how we connect the practice, and that is to latest technologies, not only maybe in classroom, but outside the classroom. And for that matter, especially in agriculture science, we have well-developed agri research farm, well-equipped computer lab, and many other facilities which can be of a great importance in research or collaborative projects which we are thinking to envisage and do very soon. Not only that much, but we have even Professor Madan's alma mater, Haryana Agriculture University, to which Professor Bahal is also having the roots. So it's not quite far, but even we have the Mahasi Dayanand University very close to us. It's hardly half an hour drive. So I think those resources can be utilized, and we are already running a lot of projects in collaboration under the guidance of Professor Bahal. And the biggest factor or the force of motivation which always keeps us on for such projects is nobody but Professor Bahal. And he's having a highly, what I should say, the scientific temperament team where he is mentoring all of them to take up such challenges and do the great work. So with all these things, I'll say that yes, you all are most welcome. We will always collaborate in such projects, whatever manner we can. Thank you.
So thank you all. We will continue our discussions through mails, and maybe that one more time we will be face to face online when we have already written something. And Achim, whatever is to be done by me, because we have to select one leader, and Achim can be rest assured that we can develop a program, and whatever is my share, you please write me. If it is compilation of all the reports, it's okay. If I have to do my part, that what experiments we can do, it is okay. And please let me know. Okay. Madan can also uh, coordinate every aspect of because he knows biological sciences, he knows computer sciences, so he can also coordinate every aspect of disease detection management work based on artificial intelligence. So one has to take the lead to compile all information, and Professor Rajam has developed a wonderful technology, and I will discuss with him how best we can uh, include his research work in the artificial intelligence work, because whatever he is doing, RNAi, that develops a product, and that product can always be tracked by artificial intelligence biosensors, because the products can be chemopathic and chemotherapeutic. So both, it is just like a dominantly and recessive. Professor Rajam, are you here?
Yes, sir.
Professor Rajam,
Sorry.
yeah, you are here.
Yeah, I'm here. Yes.
So it is both way. You have highlighted chemotherapeutic role, it could be chemopathic role as well. Okay? So these
Yeah.
substances can be tracked by artificial intelligence, and such an intelligent work being done by you can also be included in this. Okay? And I wish you all thank you the very best, and I really thank you from core of my heart that you all spared valuable time and made the webinar scientifically enriched. Thank you very much.
Thank
I
you. Thank
would
you.
like to thank Professor Sumer Singh, Vice Chancellor of Jagannath University, Professor R.K. Bahal, and Dr. N.S. Rathi to provide this platform for all of us to learn about deep learning. I would like to thank all the speakers, participants, and all others who have supported us in organizing this webinar. Thank you all.
Thank you.
Namaskar.
Thank you.
Namaskar.
Namaste.
Okay.
Thank you.
Namaskar.
Behal Saab, if you send us
Namaste.
one message, just introducing everybody